better online. So I work in cell biology. Are you a presenter? So you're not the worker, right? Okay, I don't know everybody else. I love you. I'm split molecular and cellular biology and bioengineering. So, I'm in DCL, and there's a big
Good morning.
Outreach like this.
And I'm assisting with the organization of this Biosensing Bioactivation Summer Institute 2010. And I'd like to invite my colleague, Professor Jimmy Shah, who is the chair of the Summer Institute, and Professor of Mechanical Science and Engineering, to chair this plenary session. Jimmy Shah. Thank you.
Before I start, well, welcome. The first thing, I think, I know some of you came a long way, all the way from Taiwan. And last night I had dinner with many of you, and it's not easy because dinner over here was your breakfast, but you ate well. I hope you slept well last night. And from now on until July 23rd, for the next 2 weeks, we'll be working hard. And before we start, it's my great pleasure to introduce to you Dean of Engineering of University of Illinois, Urbana-Champaign, Professor Edison.
Thank you. Good morning. Thank you, Jimmy. As Jimmy said, many of you have come from far and wide, from Taiwan. There's somebody from Marseille this morning. Thank you for coming here. We appreciate partnerships, international partnerships within the country. And we thank our partners in Taiwan very much for coming. I've been here for a long time at the university. I think you've come to a great place. And I think for the next 2 weeks, as Jimmy said, you work very hard. Because hard work in Champaign, you see, we don't have too much to do here, so we work very hard. I'm sure by the time you go around, you see a lot of cornfields and soybean. After we finish planting corn and tending to them, we come to the lab. So you enjoy the lab here. You see that there are many great resources on campus, in the lab here and elsewhere. So you have a great opportunity to interact among yourselves, because you're the leaders of the future. So it's high time and a good time to know each other, because as you move ahead in your career, these are the people you'll be talking to since you're in the same area, and people you'll be interacting with at conferences. They are great places to meet people. And you'll meet a lot of professors here over the next 2 weeks, and we'll teach you a lot of new things. And this is a new area relatively on our campus. I think Professor Jimmy Shah, Rashid Tasher, and Ifan have brought this to life over the last many years. So you'll find that there are great progress in this field, that and great challenges and opportunities that the young ones should be looking to and working to discover new things that will change science, that will change engineering, and essentially that will change society. Because above all, I always say that engineers bring good things to life. Because without engineering, you will not be able to change the human condition. So all the things that people enjoy today, whether it's your PDA in your pocket, the big computers that people use, you find that they are the toils and the toll of engineering. So the things that we'll be discussing here over the next 2 weeks, teaching you, learning, that we'll be contributing to, I think they form the tapestry and the basis for the future. So and engineering is about the future, really. Engineering is about the future. Because you ask yourself questions, what can I do today that will change things in the future? So don't forget that. Don't forget that engineering is about the future. So whatever you do today, you have consequences. So you think very carefully about what you do today, because it will have consequences in the future. So I don't want to waste your time, and let you get to your hard work very fast. Thank you for coming. Don't forget though, within all the hard work, enjoy yourselves. And don't forget to visit the campus. This is a lovely campus, a big campus, but lovely. On the north side is engineering, on the south side is everything else, but we all work across the university. The professor of education sitting at the back, so we work very collectively on campus.
Far.
Yeah, because education is Professor Design, the Stefano, so we work very closely amongst ourselves on the campus, because that's the only way we can move a big enterprise like this into the future. Thank you for coming, and again, welcome. I don't know how to say welcome in Chinese, but I always say Wan Yi. I know Ni Hao and they say so. Welcome, Wan Yi, and thank you very much.
Thank you very much, Dean Adista. For those of you who don't know, actually, College of Engineering over here is one of the largest colleges on this campus. And it turned out that the engineering on this campus is one of the best in the country and also in the world. So Dean of Engineering is a very powerful person. Be nice to him. Now, before we start, a few things I want to sort of introduce to you as the to put things into perspective. One of the things, well, this is our NSF, NSC, some Institute of Biosensing, Bioactuation, but this is not an isolated event. There are some other activities going on campus related to this. I'll go through that. I'll tell you why I'm talking about these. One of the activities is a new NSF STC, or Science and Technology Center, called Emergent Behavior of Integrated Cellular Systems, or EBITS. And the STCs are by far the largest NSF research center support. It's $25 million for 5 years. And this particular STC will start on August 1st, 2010. So in about a month or so, or 3 weeks, officially the STC will start. Just be aware of that. And the PI is Professor Roger Kent at MIT. It's a collaborative effort among 3 institutions. I'm the local lead at UIUC. Bob Naren at Georgia Tech is the Georgia Tech lead. And the idea of the STC is to gain a deeper understanding of how cooperative cell behavior would lead to formation of large organized cellular systems. And essentially, or ultimately, what we want to do is to create biological machines with multiple cell types that can perform specific functions. Okay. This is one activity. Another activity we have just received, this is not final official yet, but on the NSF website it says recommended. This is IGERT. IGERT stands for Integrative Graduate Education Research Training. This is another center grant. Rashid Bashar, who's there, sitting in the back, I'm going to introduce him, is the PI for this. Several of us are co-PIs. And the goal of that is to produce the next generation of research and education leaders. And again, this is in the area of collaborative effort between engineering and biology. Okay. When Dean Adisada gave his welcome remarks, he said engineering, engineering, engineering all the time because he's Dean of Engineering. But we know that in the audience there are maybe 30% of the students are actually from biology or chemistry or physics or other sciences. There's actually a pharmaceutical science, one student majoring in pharmaceutical sciences. So it's all about collaborative effort between engineering and biology. So it's extremely important. They see this as strategically important, as an area that we want to pursue. Okay. How to do that? Train the next generation of researchers. I wish I had the opportunity to take a course like this when I was in school. But this is your chance. Do it, and hopefully we will start to collaborate in many of these areas. Another new grant we will receive is an NIH or National Cancer Institute training grant. Again, Rashid Bashar and Natalie are two co-PIs for this training grant. This is also for training the next generation of leaders, research leaders and educational leaders in the area of nanotechnology in cancer. Another thing I want to mention is this global enterprise for micromechanics and molecular medicine, or GEM4. GEM4 is global enterprise for micromechanics, molecular medicine. That's M to the 4th power. And that was established by Subra Suresh, who is Dean of Engineering at MIT, or current Dean of Engineering at MIT. He was just nominated by President Obama to be the next director of NSF, actually. So he's going through the Senate confirmation process. The GEM4 is an organization started in 2005. Professor Suresh was the founding director. The immediate past director is Professor Roger Kent. I'm the current director of GEM4. And one of the activities GEM4 organizes is a summer school series. And you can see all these summer schools organized at different places. The reason I'm talking about this is they're all related. All these things have one component in common, which is the global outreach. This is the map for the Eiger. You can see that the center of the universe is shunting around. For the Eiger program, we have all these different partners. International partners include Cambridge University, Max Planck Institute in Europe, and Tsinghua University, National University of Singapore in Asia, and all these other partners. We actually have one student from UC Merced, which is our minority serving partner institution, and we have several students from North Carolina Central University, which is another partner we have. Okay, so welcome to you all. Now, going back to our Summer Institute, let me put it into perspective. This is something that we're going to do for the next many years, hopefully. This year is the inaugural Summer Institute. That's here. Next year, we're going to have the next Summer Institute at National Taiwan University, and we're going to rotate between Illinois and Taiwan, National Taiwan University. The objective of the Summer Institute is to provide a platform for researchers from a diverse background, in particularly particularly in engineering and biological sciences, to exchange ideas in biosensing, bioactuation area. Eventually, we hope to train these interdisciplinary researchers who are knowledgeable in both engineering and biology. And if you certainly we want to acknowledge our sponsors, National Science Foundation in the US and the National Science Council in Taiwan. They are co-sponsoring this event. And locally, for this particular event in 2010, these are the organizations that are co-sponsoring this event. Now, who are the people who are attending this? That's you. You can see roughly about, let's say, 45% from Taiwan, 45% from US, and there are a couple of them from India, Pakistan, Colombia. Okay, so this is people from the world, and the majority of them are from engineering. We have about 47, 48 students for this summer institute, and 30 plus are from engineering. That's not surprising. And we have more than 10 from biology, and we have 5 to 6 from chemistry, physics, and other sciences. Among the students in engineering, about 40-some% just mentioned engineering, non-specific. They didn't really write down what engineering discipline they're from. Roughly about 20-some% are from mechanical engineering. And they're from another 10 or so, 15% from electrical, computer engineering, and there are people from chemical engineering, bioengineering, material science engineering. So this is you. And who are we? The people who are organizing this, lecturers. You're going to see people coming in and giving lectures. I don't have to introduce them. You'll meet them. Take the opportunity. These are experts in their respective fields. Take the opportunity to introduce yourself, ask them tough questions, and develop some collaborative effort, or get to know them well during these 2 weeks. Lab instructors, I'd like to, if they are around, I'd like to introduce them. Joanne and Nestor. John Hughes. Joanne is in charge of the cell biology lab. John is in charge of the macro-nanofabrication lab. Larry Miller, oh, just came in, just in time, okay. Larry is doing the enabling technology lab, microfluidics, and so on. And Tony Griffin. He's not here yet, okay. And other people I want to introduce, program assistant, important person, Atif. And we have the program evaluation specialist, who is Professor Roseanne Stefano. She's going to help us with the program evaluation. She will tell you what to do. And then finally, the organizing committee, myself, and Irfan Ahmed, Rashid Bashar, and Tahir Saif is in Singapore right now, so he's not here. And I will let our Taiwanese colleagues to introduce themselves. But before I do that, the Chinese tradition is when you come here, you give a little present to the host, and it turned out that they brought me a little bag. And one thing I want to share with one particular person who's done so much, who didn't sleep until about 2 o'clock in the morning, sending out emails Saturday evening, Saturday night, or early Sunday morning, and then went onto a bus at 3 o'clock in the morning to go to pick up the Taiwanese people. I'm going to give that to you.
Oh, thank you.
Okay, so now it's my great pleasure to introduce Professor Cho-Hung Chang, who's the PI for this grant in Taiwan. So he received the National Science Council Award. I received the National Science Foundation Award. So let's give him a big hand.
Well, Jimmy and many your team members and all the DSBA Summer Institute delegations. As the PI from Taiwan, and I'm very grateful for both sides of National Science Council and National Science Foundation. Because of the NSF strong support, so therefore the NSC in Taiwan also give the very supportive award, which is more than I expect. When I submit the proposal with Jimmy, I expect 3 years support for this particular program, and at the end of the last year, I received the formal support for 6 years program, so which is we cannot get away from this for at least 6 years. And first of all, I want to thank Jimmy and your whole team for a great effort. Because when I look at your website and look at this tremendous work, so I said, wow, what I'm going to do next year. Yeah, we wish. And yesterday when we arrived, and our team already waiting in the airport, so we just made me very excited and so appreciated. Okay, and then after we arrive, and then we look at our dorm and look at the hotel, which is very good. Right? As far as I understand, I think all our delegates, our students are quite happy so far. Right? So, and our delegation has a total of 23 members, includes myself and Professor Xia, Professor Xiao, which is our Secretary General. And our 23 people, we have almost, we expected to have a half student from the biology or medicine side, and another half from science and engineering side. But as a matter of fact, when we received the application, which is over 100 applications in the whole Taiwan, and from there we only choose 20. And finally, we select 3 junior faculties and 4 postdocs, and I think it's 11 PhD students, and the rest 4 are master students. And we're also very happy that we have 1 delegate from its R&D oriented organization. It's called ITRI. I don't know, some of you may have heard of it. He's sitting here. We're also happy with that. And the other thing is finally we recognize that we have 8 female students, which is also make our group very happy. All right, Jimmy just told that next year will be held in NTU, which is not too far from here. So it will be in summertime as well. So we welcome you to come to Taiwan, and then the following year we'll be back to here. Okay, we thank you again, and we wish we all be successful summer institutes, and thank you very much. Okay, maybe we can, allow me to present you the Secretary General. It's a Professor Xiao Yeah, he's from NTU Mechanical Engineering Department, and he is the person who did all of this work. He also prepared the appreciation plaque to present it to our partnership here at the UIUC. Maybe this is a good time. And also, he also designed a T-shirt. And also, I'd like to give it to our counterpart students and team members as well. Okay, so maybe it's time to do it here now. Really like to appreciate Jimmy for his excellent organization and everything, all the program he put it together.
It hasn't started yet.
Oh, thanks so much. Thank you. Thank you. Great job. Present to you as a whole.
Wow, I could put it on.
There's FNSC, some institutes in the front and in the back.
Every one of you will get one?
Yes.
Okay, so we'll go out and, high school students, right?
Please come to me during the break.
Okay,
I'll give you
all right.
feedback.
Thank you very much. Wow. Yeah, we're going to have fun even before we start. All right, some logistics, important things. The first thing, food. Breakfast and lunches will be, and coffee breaks will be provided. Dinner, you're on your own. Well, we turned it positive. Dinner is for you to explore the beautiful Champion Event area. Internet connection. All of you who are from places other than U of I, on the first front page, you have a little card which shows your internet connection. And one of your last sheets over here tells you how to connect to the internet. If you have a laptop, follow that instruction to connect to the internet. Lectures will be here, so this will be your home. This room, 1000 MMPL, will be your home for the next 2 weeks. You will eat here, you will take lectures here. You don't live here. Labs, you have been assigned for a particular lab tool. It's on the front page. It's color coded. Okay, so your tool B, your sort of a reddish color, and it's also on the name tag. Name tag is color coded. look at the name tag, you have the color of the group over there. And they will, the Atif and other lab instructors will lead you to the lab location. Evaluation, maybe this is time for me to turn it over to the thing. And you have a little form on the very last page, insert over here. This is the pre-institute evaluation.
Good morning, everyone. It's good to see you here. As Jimmy said, my name is Lizanne DeStefano, and I'm a professor in educational psychology. Engineering and the sciences and education work very closely together on this campus because we're very interested in improving our education programs in science and engineering. We think that activities like this, institutes like this one, are a good way to promote international relationships, start research collaborations, train the next generation of researchers. And so we are going to be studying this institute very carefully to make it better and also to learn from it as we plan forward and plan other activities. So your information is very important to us in improving this experience and also creating other experiences. That paper that Dr. Shaw just showed you, the evaluation form, we would like you to fill that out today and turn it into...
By lunch?
By lunch.
On the registration desk outside.
To the registration desk outside.
Okay.
And then we will take that, I will compile, put together all of the findings, and the team will meet and go over those findings and think about, well, how can we do things differently? You see on your agenda, it says schedule is subject to change, and so we may decide to do things differently based on your feedback. There will also be people interviewing you throughout the institute. They'll identify themselves as evaluators, and you should feel free to talk to them about your experience, what's going well, what you think could be improved. And then at the end of the session, we'll have another evaluation so that you can reflect on your experiences. And once you go back to your institutions, we will have another evaluation to say, well, are you using anything that you learned at this institute? Because sometimes we find that those evaluations that you fill out right at the end when you're going home tell you one thing, but then when you go home and you're thinking about the experience, you actually feel differently about it. So don't be surprised if you keep hearing from us. We want to stay in touch with you, but it's very, very important that you fill out the form and turn it in by lunchtime. One other thing is that you will not be identified in any way on the form. When I give back the results, I will not mention it by name or anything like that. There is a space for your name on the form, but that's only if you want to identify yourself. You can turn in the form with no name as well. Okay? If you have any questions, I'll be around. You can come up and ask me them. Okay? Thank you.
This is so important, as Lizanne said. So important, so if you don't turn it in, you don't have lunch. All right, we're going to take a group picture on Thursday at 10 o'clock in the morning, so be prepared for that. Bring your best appearance. There's going to be barbecue Thursday evening, this Thursday evening, at the Crystal Lake Park. Since World Cup just finished, so we think maybe we have a local World Cup, the Taiwanese team versus the international team, or maybe some of these. And there's a little lake, you can go boating, fishing, certainly relaxing. Okay? We're going to have a research idea exchange this Friday. We call this speed dating style. Any of you have participated in speed dating? No? Some of them. Yeah. Yes. Okay, so essentially what you do is you form pairs, you talk to each other for 5 minutes, then you rotate, you change your partner. Okay? And hopefully some of these interactions or exchanges will result in long-term collaboration. Research collaboration. Maybe some others.
Yeah, right.
Who knows? But it's all fun, no pressure. We're going to have an excursion on Saturday. We have a bus that will bus you to Chicago to have a one-day tour on Saturday. Please sign up on the sign-in sheet. Right, you have that. Okay. If you haven't done that, go to the registration desk. We're having a poster session next Thursday. The poster session will have 1 minute presentation. You can prepare 1 slide, or if you don't have 1 slide, you can just talk about your poster. The purpose of that is to attract other people to go to your poster. Then the poster will be set up upstairs on the 2nd floor, 1st floor, 3rd floor, and in the afternoon, Thursday afternoon, next Thursday, we'll have the poster competition. We'll hand out the best poster award. Then Friday, we'll have a lab tours of different places, faculty, institute, and so on. Then we'll have the graduation ceremony. Graduation, we're going to hand out a certificate signed by Professor John and myself. And then by that time, you're supposed to fill out another evaluation. And we have just decided this morning that we'll give you some other thing to do. So in addition to in order to get the certificate, graduation certificate, you need to write a short report, okay, saying what you've learned and your experiences over here and what you're planning to do. Right? Okay. That's it. Questions? Go to the website and let the Summer Institute be. Thank you. Now, we'll give Professor Bashir a few minutes to talk about this place. The micro nanotechnology.
All right, thank you, Jimmy. So I'd like to also welcome everyone. My name is Joshua Bashir. I'm a professor in computer engineering and bioengineering. I'll keep my comments really short just to keep us on schedule. But the main purpose of my being here is just to welcome you all. I want to first of all thank Jimmy Shah, Professor Irfan Ahmed, and Professor Chang for all their hard work, and also Atif for putting this wonderful program together. Again, I think till you go through the exercise of putting something like this together, you don't appreciate how much work it is. And I think our collaborators from Taiwan also know this. So there's a lot of world-class speakers coming in the next few weeks. It's a great opportunity. So I really, would highly encourage you to make the most out of it. I think we're going to keep you busy, very busy. The lunch is even only 30 to 40 minutes, so it's going to be a really tight schedule. But just enjoy that and learn as much as you can. Right now, you are in the micro nanotechnology lab. This is one of the 6 or 7, depending how you count, centers on campus that are interdisciplinary. We have interdisciplinary research facilities, which the purpose is to help facilitate research across different departments, different colleges, and we've been doing that, working very hard on that in the last many years. We are also very, very proud of the history and the tradition and the contributions of the faculty in the nanotechnology area, in the microelectronics area, and the photonics area. I'm not sure if you all know, and hopefully through the whole process of being here in the next few weeks, you can get to learn more about the contributions of the faculty from here. But Professor John Bardeen, who is the inventor of the transistor, was here as a professor. After he invented the transistor at Bell Labs, he came over here and he was a faculty member in ECE and physics. And he hired 2 students when he came, his first 2 PhD students. One was Professor Nick Poloniak, who is credited to be the inventor of the visible LED. So the light emitting diode that you see around us, he's been credited as inventor of that, and he's actually still active and is here in this building. His office and lab is on the second floor. And the other student that Jean-Marie hired was C.T. Sa, who is credited as a co-inventor of CMOS. He was here for many years and he moved on to Florida. So there's tremendous history of and contributions of electronics and photonics. And then also in the last many years or so, we've also in this building really pushed a major focus in the bio area. So we're also trying to integrate the biomedical sciences, biology and medicine, and trying to integrate that with engineering. Because certainly healthcare, the delivery of healthcare, and the basic understanding of biology, we now say that biology is the next frontier for engineering. So there's a lot of interest at the nexus of engineering and biology, and that's really one example of this workshop. So we'll be doing this actually every year, either through our collaborators with Taiwan, and also here through under the auspices of the 2 grants that you heard about, the 3 grants actually, the NSF STC, the NSF IGERT, and the NIH training grant. So hopefully we'll have this as an annual workshop, so you also should go and tell all your friends about that we'll be doing this, and encourage them to apply for the next many years. And again, just have a great time here. And if you have any questions and any comments, then you can contact us. And hopefully through the next few weeks, you'll also get to see this facility. You'll visit the clean room and the various labs. So we don't have an official tour for MNTL because you're going to be seeing the labs anyway. And actually, I think next you're going to see a short video about MNTL. So again, thank you all for coming, and I also want to congratulate you for being here. This was a very select group. So you should actually be proud of being here. All right, thank you.
For those of you who don't know what Jean Bardeen looked like, his picture is outside, so take a look at that when you go out. Another thing I think I should add is Jean Bardeen is the only person in the history of the world who got 2 Nobel Physics Prizes. There are people, Madame Curie actually got 2 Nobel Prizes, but one in physics, one in chemistry. Bardeen is the only person who got 2 physics Nobel Prizes. The second one actually, he did the work over here at the University of Illinois on superconducting. All right, let me introduce to you another person who played a major role in the success or in the organization of this Summer Institute, Dr. Ahmed. Okay. He's the Associate Director of Center for Nanoscale Science and Technology over here.
Thank you, Professor Dinisha, and welcome. On behalf of the Center for Nanoscale Science and Technology, which collaborates the entire nanotechnology on campus, including the labs that you are in, Micro Nanotechnology Lab and others, is a virtual center which is supposed to bring in research, education, outreach, and entrepreneurship, and that connects the university as a whole to the outside world with 150-some researchers engaged in micro and nanotechnology research. Now we are going to play a video clip which highlights some of the activities within the micro and nanotechnology lab, and you have a good sense. And so give me a second to bring that up. And we can get the lights there with you, please.
excellence and leadership with paradigm-shifting technologies leading to multi-billion-dollar industries such as the transistor, semiconductor materials, the LED, plasma displays, and a host of other novel discoveries. All of these strengths of the MNTL enable its researchers to work on important societal problems and issues such as healthcare, energy, the environment, and agriculture, leading to new areas such as nanomedicine and bio-nanotechnology. Recent research thrusts include micro and nanoelectronics, nanophotonics and optoelectronics, MEMS, NEMS, and integrated systems, and nanomedicine and bio-nanotechnology. The Micro and Nanotechnology Laboratory is well positioned to conduct cutting-edge micro and nanotechnology research, and has also trained the next-generation workforce to maintain the state's and nation's leadership in science and technology, as well as its economic prosperity.
The MNTL builds on a strong tradition in the areas of physics, materials, and electronics, and that started with John Bardeen and in Colonia. And of course, now it provides opportunities for our faculty and students to continue in that line of work.
Owes its existence to semiconductor, owes its existence to the old man, to the grand old man, one of the greatest physicists ever, two Nobel Prizes in physics. Only person with two Nobel Prizes in physics. There at Princeton, where Bardeen got his PhD, was where Seitz just ahead of him got a PhD. Seitz was a little younger. But they were the beginners of the solid-state physics in the U.S. And at some point at the end of the war, Loomis hired Seitz, who brought solid-state physics to Illinois, got the dean to make an offer to Bardeen. Bardeen was the guy that brought semiconductor work here in 1952 and in 1951, and in 52 started the laboratory. So semiconductor work has been going on at this school since just after the invention of the transistor, brought here by John Bardeen from Bell Labs. And then Bardeen spent the rest of his life here, 40 years, working on superconductors, many times.
The most commonly used technology for quantum well laser was invented in 1977 when I was a graduate student here. And that laser, quantum well laser, is being used by everyone in the world for application, especially technology very certain. Then we also have another professor, C.P. Sa, who invented CMOS. So all the integrated circuit that is using a CMOS technology. We made the world's fastest transistor. And, since 2003, 2004, and up to now, it's still the world's fastest transistor.
The Micro and Nanotechnology Lab has a history that is unmatched by anybody, anywhere, because of the continuous contribution by well-known people in the field, from Bardeen to Bologna, CTSA, Greg Stillman, and the present faculty in the Micro and Nanotechnology Lab. So that's a big history for us, and it's something that we are very proud of, that has originated in the credit of the United States.
I don't know any school has that longevity and that much history. Micro is a direct product of the semiconductor, what the semiconductor is. It's just tiny little stuff at the beginning. And vacuum tube people and ordinary electronics people laugh. They didn't think they could amount to anything. It changed the world.
The Micro and Nanotechnology Laboratory is providing an environment including, of course, facilities and offices for students and faculty to perform work in the area of electronics, nanoelectronics, materials, and devices which will help us as an institution create solutions for very critical health and energy problems. Health and wellness, energy, and the environmental sustainability are critical areas in our strategic plan, and as a result, MNTL is contributing in great ways to promoting the objectives of our
But the Micro and Nanotechnology Lab currently is the home to the largest clean room on campus, and it serves users across the campus and actually across the state and across the country. We also have a new bio-nanotechnology lab that has been set up within the Micro Nanotechnology Laboratory that's also available to
very early work on a process for string layer lasers that has had a significant impact on the industry. And subsequently, we've been working on things like selective area epitaxy for photonic integrated circuits. And more recently on quantum dots and other reduced dimensionality structures that have some features that are not commercially haven't had a commercial impact yet, but most definitely will in semiconductor field.
MMPO is the building which has all the necessary equipment to allow us to fabricate this world battery system, and also allow us to build the current generation and next generation the integrated circuit technology. That going to be extremely important. Is the roots of what we're going to do. All the dimension we're talking about is already in the nano scale. What really got us to know now is being translated for the areas of science, and in particular, we call it technology. Now we call it nanotechnology, which covers the full spectrum of technology. That we now go into medicine, or how to look at technology and use to investigate fundamental principles of knowledge that nobody else is able to.
We have the opportunity now to do work really at the forefront of what's called bio-nanotechnology now. We're developing technologies that are used in pharmaceutical research, in healthcare, things that are used for treating diseases. We're building everything from highly sensitive sensors for DNA molecules and proteins to things like artificial retinas and tissue engineered structures for replacing tissues in our body or parts of organs.
We have the faculty across the college that uses micro and nanotechnology as a facility. These are faculty we've been recruited over the last few years, and we're going to get more into this area. This is a public facility.
So the future role for MNPL is reflected in the addition of the building. We've expanded our physical plant, but we've also expanded the range of tools that we have available now for research.
With all of the tools and technologies that we have here, we have the capabilities to build almost anything that we can dream of in terms of compound semiconductor structures, high-speed transistors, lasers, LEDs, sensors. But then we can also interface those things with biological stuff. So we can take those kinds of devices and utilize them for DNA detection, for detecting proteins in blood for diagnostic tests, developing tools that can be used for environmental monitoring, and things that can be used to address pressing concerns in energy research, with things such as solar cells, things that can harvest light more effectively than technologies that are out there today.
The micro and nanotechnology will be a place where all these people with different specialties so this lab will be an integrated lab. The concept of the faculty together.
Where can this area has the potential to provide us with knowledge and devices that provide solutions in the energy and health areas? And these are problems that we are trying to solve as a university. Obviously problems that if we can make advancement in their solution, we can put them in the time.
Our biggest contribution ultimately is going to be the students that work here. They're being exposed to high-level science and high-level engineering in a large group of distinguished faculty at all points in their careers, and access to the best tools.
Micro nanotechnology is the tool and bringing people together to write large center grants and then federally funded centers. We hope that we'll continue that in the future and continue to work with collaborators at the Institute of Genetic Biology, at the Beckman Institute of Materials Research lab, and also the organic science lab, and of course, many of the various departments to bring faculty together to work in the area of nanotechnology. We also very much hope to continue to work with industry and continue to have that very active and real interaction with industry in terms of doing research that is very relevant to industry.
I think that's it. Okay, so yeah, we are ready for a
So we are very grateful that Micro Nanotechnology Lab could provide the facility for us. We are running slightly behind schedule, as usual. However, we want to give all of you a chance to introduce yourselves. So let's do that. Just tell us your name, where you're from, why you are here.
My name is Farhan. I'm an undergraduate student at National Institute of Technology. This is something that's very relevant to me. My major is in instrumentation and control, which is sensing and actuation translated. And I want to see how the bio component can really make a difference.
I'll give you at most 15 seconds for each person.
Good morning. My name is Chen Li, and I come from National University, Taiwan. And I just received my PhD degree this year, and now I'm a postdoctoral researcher in National University. And my research interests include reliability design for integrated circuit and the circuit design for bio application. So I come here for study about bio application.
Hello everyone, my name is Yu Huang, and I come from Taiwan University. And I major in electrical circuit design. So I'm looking for biomedical application to such a combination of the sensor and actuator with my circuit design.
Good morning, everyone. My name is Zhong Jianwu, and I am currently a PhD student in National University, Taiwan. And my study was working on physical and signal transmission of the cells under synthesis and engineering procedure. And I try to quantify the mechanical property of the cells. And thank you.
Good morning, everyone. My name is Shizhong Wei. You can call me John because it sounds like Zhong. So I come from National Taiwan University, and my research is about biosensor because my background is medical technology. So I'm interested in detection or sensing about DNA, protein, or cells. Thank you.
Good morning, everyone. My name is Chin Tong Chen, and or you can call me Jimmy. And
He was named after me.
And I come from Taiwan, and I'm now a PhD student. And my research is focused on the biosensor development. And I come here to learn more about other people. Thank you.
Hello everyone, my name is Qian Chen, and you can call me Annie Chen. And I'm from Taiwan University, National Taiwan University, and I'm now in my first year master program. My major is biochemistry, and I'm now working in academia Sinica. And our lab is the main project in our lab is to produce fluorescence nano diamonds to do some kind of bio experiments. Thanks.
My name is Jesus Luna. I'm from the University of California at Merced. And I'm here because I want to learn new techniques to apply to my stem cells. I'm doing a project involving microfabrication in stem cells.
Hi, my name is Daniel Kim. I'm a PhD student here at UIUC. My current project is related to measurement of cell mass. Because my major is mechanical engineering, I want to get some basic knowledge in biology and cell mechanics.
Hello everyone, I'm Yin Fang Chen. I'm a young fresh PhD from the National University. My research interest has been focused on how ion transport system cooperate with the cell plasma membrane receptor and thus influence the intracellular signal. So this is too complicated to affect the intracytoskeleton system and thus influence the cell invasion and cancer metastasis. So why I'm here is I want to integrate what I will learn here, something about biosensing and biointegration, integrate what I will learn into my future research. I want to further study how the system I mentioned above that will affect the cell force or cell migration.
Everyone, my name is Zhou Wenbin from Taiwan. My BS degree is botany, a green science, green kind science. My master degree is molecular medicine, and now I study in mechanics engineering for a PhD degree. Yeah. So I'm very interested in about any new technology, including biotechnology and new engineering. So that's why I'm here. Thank you.
Hi everyone, my name is Ruth Lancheros. I am a student, a PhD student now at the Universidad Nacional in Colombia. I'm from Colombia. And I'm here to learn more about this because my focus is on nanotechnology. I want to make nanoparticles to drug deliver. So thank you.
Hi everyone, my name is Yola, and I'm a first year PhD student in Department of Environmental Engineering here in the UFI. And my research is mainly on the mechanical property of the tissue. And I'm here because I want to explore myself and use data for biosensing and all of you guys. Thank you.
Hello everyone, my name is Jiyo Lai, and you can call me Grace Lai. And I'm a PhD student from NTU, and my major is in immunology. And my research is trying to find biomarkers for schizophrenia disease. Thank you.
Hi everyone, I'm from North Carolina Central University. This is my CV and master's degree at pharmaceutical science. My research is on how to, it's about the treatment of cells by carbon nanotube, and very interested in this technology and learn how this biosensors detect and monitor the biology activities.
Hi everybody, my name is Dana Austin. I am a second semester graduate student at North Carolina Central University, and my research is determining the role of cortisols in pancreatic cancer.
Good morning, my name is Jeray Bauer. I'm also a student at North Carolina Central University. I'm going into my second year as a master's student there in chemistry. My research is pretty much dealing with drug delivery and using polymers for drug delivery. So, of course, cell studies as far as drug delivery is very important to us here.
Hello everyone, my name is Chen Yu Fu, and you can call me Chen Chinese. And I'm
study biology problem. Also have an interest in tissue engineering. Thank you.
I'm Ying-Chu Liao. I'm an assistant professor in National Taiwan University. My research area focuses on the facial science and colorists. I'm here to learn newest technology. Thank you.
Okay, my name is Pei Chen. I am assistant professor in mechanical engineering in National Taiwan University, and I do research on microfuel cells. So honestly, I have very limited knowledge about biology. So I'm here to see what my fuel cell can do to a cell, whether to maybe kill it or make it live happier. So I'm very looking forward to working with you all. Thank you.
Hello everybody, I'm a PhD student from the Taiwan University. I major in electronic engineering, and my research is about to use carbon nanotube as a cancer for fuel cell. I think I can learn a lot about biology technology and use to my study. Thank you.
Hello everyone, my name is Marco, and I'm a PhD student in the NTU. I'm studying the mechanical engineering department, and my research is about the carbon nanocoil. It's a helical shape of the nanotubes, and I want to connect this material into the biologists. So that's why I'm here. Thank you very much.
Hello everyone, my name is Shunai Huang, and you can call me Leila. And I'm the student in PhD in Institute of Physiology, National Yangming University. And I'm studying about the animal model and primary culture. And my research is LPS effects in the adrenal gland response. So I'm glad to enjoy this summer institute, and I hope I can learn a different field in this course. Thank you.
Hello everyone, my name is Zhengchang Liu. I come from National Taiwan University. I'm a master's student of Graduate Institute of Photonics. My research is about terahertz imaging and terahertz fiber. I am very happy to participate in this program because I can learn lots of cutting-edge biosensing techniques, and I can make friends with excellent students around the world. So I really appreciate the opportunity. Oh, by the way, I love playing tennis very much. Actually, I have brought my tennis racket from Taiwan. So if we have free time, we can go to tennis court and have a good game. Thank you.
We have somebody called Serena Williams.
Hello everyone, my name is Pei Hong Chen. I'm from Taiwan, and I'm doing research in bioinformatics in National Yangming University. My major is microbiology and immunology in my PhD program. But now I'm interested in genome analysis and genome design and engineering. Thank you.
Hi everyone, my name is Wesley Wang. I'm a junior faculty of National Tsinghua University in Taiwan. And my research interest is in tissue engineering in the field of regenerative medicine. So very glad to be here learning with you.
Hello, I'm Jian Chu. I got a PhD degree from National Chongdong University. My important task here is search a technology what you develop can be transferred to industry. So my interest is develop microfluidics for ultra-stress biomarker for early disease diagnosis. Thank you.
My name is Liz Brogan. I'm a PhD student here at the University of Illinois. And my research interests are in biosensors using new thin film diamond technology. And I really want to learn the biology behind the actual sensing.
Good morning, everyone. My name is Wenjuan Shi, and you can call me Jasmine. Now I'm a master's student in the Department of Pharmacology,
National University from Taiwan. And now my interest is in cytoskeleton dynamics and cytoskeleton associated proteins in normal and cancer cells. And that's all. Thank you.
Hello everyone. My name is Adil Ahmed. I'm a PhD student in electrical engineering at University of Illinois. So my research focuses on using magnetic nanoparticles for bioimaging and drug delivery applications.
I have a MD background and am now currently still working in hospital in the field of internal medicine and infectious disease. I'm now a graduate student in National University. My current project is building a decision support system to help better shoot glycemic control in diabetic patients. And it's very important.
Hi, my name is Chris Liu. I'm a graduate student here in Illinois, and my research is on using microfluidic devices to create your own cell posters.
Hi everyone, my name is Adoko Ford. I'm an undergrad from Purdue University, majoring in biology, and I'm assisting in research here at the University of Illinois relating to white blood cell detection, and hopefully I can learn some material through that research.
It's very nice to meet you here. My name is Do Qi. I'm an associate professor in Beijing University of Aeronautics and Astronautics in Beijing, China. Now I'm a visiting scholar of Professor Jun Xia, and my interest in research now in the mechanics of electronics, stretchable electronics. Yeah. Why I'm here is two reasons. The first is Professor asked me come here. And the second is I'm very interested in the biosensing and bioactuator in engineering applications. Thank you.
I offered you.
My name is Greg Damhorst. I'll be an MD PhD student in bioengineering here at Illinois, starting in the fall. My background is in physics, so I have a lot to learn about biosensing, which is why I'm here.
Hello everyone, I'm Raj Iyer. I'm a graduate PhD student here at Illinois, and my research interests are microfluidics and using the advanced technology, microfluidics technology to study neuronal development. And I'm here to polish up my knowledge in engineering, which I do not have much of, and hopefully help that in my research.
Hi everyone, I'm Moy Hassan and I'm an electrical engineer. Currently I'm working as a lab engineer instructor at Lund School of Science and Engineering, and my research focuses on biophotonics and biomedical instrumentation. Thank you.
My name is Wu. I'm a graduate student of mechanical engineering here at the university, and my research interest is to develop new learning methods to image and analyze cell dynamics, especially the transport dynamics in cell cells.
Okay, thank you all very much. Now, first test. What is his name? Forget it. It's my great pleasure to introduce my friend who's going to tell us all about biology. Dr. Heaton, Scott Heaton, who got his PhD from cell and molecular biology from the University of Illinois. I got to know him, became friends when we attended the very first Gen4 summer school in Boston. Right, right. And so he's going to introduce us to basics of biology. Thank you. Thank you, Jay.
So, as I'm sure you all realize what Jimmy just said, that I'm going to teach you everything there is about biology, that was a joke. Please be aware that that is a joke. I'm going to do my very best in the little time we have together today and again tomorrow to give you a whirlwind, which means very fast, overview of some of the very basics that we know about cellular and molecular biology. Of course, I was very gratified to hear you all introducing yourself and hear many of you say that you came here because you needed to learn a little bit more about biology in order to expand your research as much as possible. I'm very honored to be a part of that process. And my first thing I really want to say to the organizers, thank you so much for asking me here. It's a true honor and a pleasure, and I'm happy to do it. For those of you who come from a biology background and need to bolster your understanding of physics and engineering, good night. Have a nice nap. Most of this stuff that I'm going to be talking about are things that are really just the very basics of biology. And if there's anything, I'll do my very best to answer any questions you have over the next hour or two. If there's anything that I can't answer or we don't have time to get answered, my biggest suggestion is to go to your university and take introductory biology 3 semesters. I'm going to try to cover in the next 3 hours, right, or today and tomorrow, what would take 3 semesters in a biology classroom, and you would learn a lot more detail, a lot more depth. My introduction to this was in my PhD research. I was very interested to hear some of the people saying they were interested in the mechanical effects on neurons. I was looking at literally the mechanical effects, how that changes, how neurons, these are Drosophila neurons, how that behaves using these MEMS devices in the lab of Tahir Saif, who's unfortunately not with us today. So that's how I got interested in this mechanical engineering and biology mashup issue, was through my own research, and that was very fruitful. And I think it's a great thing you guys are here to do, and I think it's going to be a very fruitful avenue of research for both engineers and biologists. I'm very excited about it. Okay. Questions before I get really into stuff? Things you want to talk about? Let me talk a minute, and then we'll look at questions. Okay? Let's see here. I hope that this works. How does this advance? I guess we'll do this. Okay. So when I was searching through the web to look for images of what's going on with biology, I'm a little sorry that my first slide here, to show any actual information, is a cartoon representation of a cell instead of an actual cell. Okay? But this shows a lot of things, and this is fairly typical of the type of image you get when you start talking about cells. And one thing I noticed about this when I was trying to decide how to explain all this stuff is, I want to start off with the fact that if you're going to study all of biology, you can study biology for a long, long time and end up talking about 4 different types of molecules. Okay, 4 major categories of what we call biologically important molecules. Okay, there are lipids, there are carbohydrates, there are proteins. I've never been good doing 3. Okay. Proteins, and nucleic acids. Okay? So those are the 4 major categories of biologically important molecules. And the reason that they are important molecules is because they can build, with those 4 types of molecules, they can come together in ways that build huge, giant polymers and give us the structures that we have when we look at a cell. Okay? So when we're looking at this cartoon image of a cell, one thing that struck me right away was that if we look at this, first of all, the thing that surrounds and defines the cell is called it's called the plasma membrane. Okay, the plasma membrane. All right? If we look at what surrounds and defines this area in the middle called the nucleus, and if we look at this endoplasmic reticulum, this long series of membrane inside the cell, what defines the peroxisome, which is a little organelle that's kind of like a garbage dump or a recycling bin, right? Everything that the cell wants to break down and reuse goes through this peroxisome. All of the things that surround and define these compartments of the cell are made of lipids. And we're going to get more into the details of that here in just a few minutes, all right? So lipids is one. Lipids make membranes, right, which surround and define different regions of the cell, okay? Then there are, but really, if all the cell had was membranes, engineers wouldn't be interested at all. It'd be boring as all get out. All the cell would be is a bag of water if it was just lipids, right? Because it would take on the most energetically favorable form, which is a sphere. It would contain water. It would be boring. Nothing else would happen if all there were lipids. What makes a cell interesting and functional are proteins. And we're going to spend a lot of time today and tomorrow talking about proteins, about how they're made, about what their subunits are, about a little bit about how they obtain their function. And most of the stuff you're going to hear about for the rest of the week, people are going to be talking about how interesting proteins are, how this protein affects what happens in a cell. And proteins give the cell its shape, its behavior, its function. Proteins are what make life interesting. By life, I mean literally living organisms. Okay? And so that kind of leads me to the next type of molecule, because proteins are molecules built by having a specific sequence of their building blocks, which are called amino acids. But what determines that specific sequence of amino acids is the third type of molecule, the nucleic acids, specifically DNA. I'm sure you've all seen the structure of the double helix of DNA, and you've all heard that DNA is the instructions for how to build life, right? How to build bodies. And that's true, but what we really mean by that is DNA is the instructions for how to build proteins. This is why DNA is so important, and we're going to look a little bit at that process of turning a DNA sequence into an amino acid sequence, which then folds on itself to be a protein. Okay? We'll get there. Okay? And then there are carbohydrates, and I'm not going to talk much about carbohydrates. Carbohydrates are very important for the body in terms of energy storage, as well as in terms of giving some areas of the cell some of their physical properties, some of their mechanical properties that might be really interesting, but I'm just not, and also for identification, for immune responses and so forth. So they are very important types of molecules, but I'm going to focus this week on lipids, proteins, and nucleic acids. Okay? So again, this is the cell. I talked briefly about some of these different parts of the cell, and it just struck me that so much of this picture of a cell has to do with things that are membrane, right, are lipids. But lipids are fascinating stuff. They have some really interesting mechanical properties that I talked about a little bit, but really without proteins, the cell would not be able to function. All right, so here we are. We've got all of these things which are, so the plasma membrane, nuclear envelope, endoplasmic reticulum, organelles, these are all lipids. Okay? So the lipids here, right, that's our first category of major molecule. All of these things are based on lipids. Then we've got our proteins. And our proteins include all sorts of really interesting things, right? Like the cytoskeleton that gives structure and shape and motility, movability to to the cell. Shape, force, and motion are things that proteins do. Also, chemical catalysts. We'll talk about a lot of the things that proteins do for us. And finally, there are nucleic acids. And we're going to spend most of our time talking about one nucleic acid, DNA, deoxyribonucleic acid. I'm sure you've all seen the double helical structure. And its main function is to store the information as to how to build proteins. And we're going to see how all that gets carried out. One more little thing on here on this diagram that I should point out. These little purple dots here, all over the place, are labeled as ribosomes. We're going to come back to ribosomes after a while. Ribosomes are the place where proteins are synthesized in the cell, where proteins get made. And carbohydrates I'm not going to talk about. Okay, lipids. Let's look into these guys. Again, they have several functions in the cell. As you can see, I have a few too many lipids built up within myself. Right? They are very good energy storage molecules. Okay? But they all have other, even more important functions in the body, in the cell as well. This is the general shape of a lipid. Okay? We've got two major regions. We've got this region up here, called a glycerol unit. Here we have the carbons, oxygen, and hydrogen spelled out. And then we've got these long fatty acid tails. Now I'm going to assume, how many people have had an organic chemistry class? Okay, okay, good. I'm just getting some idea of the audience. For the rest of you, organic chemists are lazy, okay, in their notation. But they're lazy in really smart ways. Okay? And one thing they want to be able to do, and they have to do, organic chemistry is the study of carbon-based molecules. One thing I maybe should have mentioned when I was talking about the four different categories of major biological molecules, I want to really pound this home, right? Lipids, carbohydrates, nucleic acids, and proteins, right? All four of those are based on carbon. Right? The atom, the element carbon. And there are good reasons for this. And we'll get to that. But mostly, I just want you to know that organic chemistry is the study of carbon. Okay, that's why it's called organic, living, right? Because all four molecules are based on carbon. Well, organic chemists get tired of writing C, 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 C. So instead, they came up with this wonderful little shorthand where all they do is make a squiggly line, and they assume that you know that every bend in that line, there should be a carbon there. Okay, carbon, 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 carbon. Okay, you get the idea. Now, one thing you'll notice about carbons when we write them out is that they make bonds with up to 4 other atoms. Right? 1, 2, 3, 4. Even when they only make connections with 3 other molecules, they still have 4 bonds. One of them just is a double bond. Okay, so this is carbon double bonded to oxygen. 1, 2, 3, 4. What's here? What should that be if we wrote the letter? C, carbon, right. So, they all want to have 4 bonds. But look at this. Let's take this carbon as an example. We're only seeing 2 bonds. One to this carbon and another to that carbon. This is how smart this organic chemistry shorthand is, and I promise I have a reason for telling you all of this, right? Not only do they assume that every bend is a C, just assume there's H's everywhere else. Hydrogen. Hydrogen is the most common element in the universe. It's everywhere all the time. It takes up all the bonds that aren't taken up by other things. So when we see this squiggly line and nothing else, this means there's a carbon here, a carbon here, a carbon here, so that's 2 bonds for this carbon, and then a hydrogen here and a hydrogen here, so 2 other hydrogens, they just don't bother writing them. Okay, so we've got these 3 long chain fatty acid tails that are carbon, 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 and that's it. Now, this means, well, we'll move on. I wish I could get this thing to advance. Okay. This is a slightly different molecule. It's also a lipid. This one is called a phospholipid. By the way, that previous one is called, you might have heard it when talking about diet, is called a triglyceride. Tri, because there are 3 long-chain fatty acid tails. Glyceride, because you've got this glycerol region, and then your 3 long-chain fatty acid tails. Okay? Now, this one is called a phospholipid. Okay? It's got 2 long-chain fatty acid tails. And then, on this 3rd carbon of the glycerol, instead of having a 3rd long-chain fatty acid tail, it's got this phosphate group. If you're not familiar with phosphate groups, you might want to write down the structure of that phosphate group, because we're going to see phosphates over and over and over again. They're really important little groupings of atoms. It's a phosphorus with 4 oxygens. Okay? Now, this phosphate group is charged in terms of how it interacts. By the way, cells, there was an old Star Trek episode where we meet some alien. And the alien looks at a human being and says, ugly bags of mostly water. That's how they've defined us. Now, I don't know about the ugly part, right? That's a matter of aesthetics. But the idea that we're bags of mostly water, spot on. That's us. We've got water in our cells, surrounding our cells. Water is a very important thing for all of life. How atoms and molecules interact with water is a major part of biochemistry. Okay? So this polar head group here, right, means this is going to interact nicely with water, because water as a molecule itself is a polar molecule. Do I have any, can I write anywhere? Can I write on here? Cool.
This one, right?
What's that?
The markers there, you might be.
Okay. Sorry, I didn't have this as a slide. Okay. Over there? Oh, okay. Good. So I'm sure you're all familiar with what H2O looks like. You've got, let's see what I'm going to use here. Red for oxygen. You've got an oxygen and 2 hydrogens. Black will do for hydrogens. All right, this is H2O, and this is a really bad drawing of what H2O really looks like. Okay, it really is asymmetric like this, and it's got 2 regions. This region up here is partially negative charge, and this region here is partially positively charged for chemical reasons that I won't go into today. We've got too much to get through. The point here is you've got one end that is partially negatively charged and another 1 or 2 ends that are partially positively charged. We call this situation polar. Okay? Now, back to our phospholipid, which is what I really want to talk about. Phospholipids have this polar or hydrophilic head region and this non-polar, very long fatty acid chains. Okay? So what this means is this part really likes to be near water, and these parts, we call them hydrophobic. What's a phobia? Fear or hate, right? It hates water. It stays away from water as much as possible. Loves water, hydrophilic. Non-polar, hates water, stays away from water. This has some really interesting ramifications. Okay? So, these, we're going to be talking about so many of these. So I already talked about what a shorthand this is for what's really going on, all the different atoms that are there that they don't show. A carbon and 2 hydrogen, carbon, 2 hydrogen, carbon, 2 hydrogen, carbon, 2 hydrogen. We are going to represent this whole thing as this little ball and 2 sticks. Okay? But every time you see this, you're going to see it a lot, I want you to be thinking, this is a phosphate group. Is this going to like water or not like water? It's going to like water. And 2 long-chain fatty acid tails, they're going to hate water. Hydrophobic. Okay? So, there's that guy. That's going to represent this whole thing. If you throw a bunch of these into water, they will spontaneously, because it's their energetically most favorable orientation, arrange themselves something like this. Okay, they will come together in these little groups called micelles, such that they produce this whole region with less water, while these head groups out here are near the water that's all surrounding that they like. Because it's energetically favorable, they will spontaneously come into something like this. Now, you have to think 3-dimensionally. This is a 2-dimensional bad representation of what's going on. Please realize this will come out of the plane of the screen and make a big sphere. Not so big, really. This is called a micelle. Okay? This is a slightly better representation, because I didn't make it, of a micelle. Okay? Notice that what's going on here is those long hydrophobic tails are here in the middle where there's not much water. They love this. As opposed to these these phosphate head groups that are sticking out here where they're going to be surrounded by water. They love it. This is energetically very favorable, spontaneous thing that happens. Okay? Now, if you add more of these, what they end up doing, and you have to adjust your conditions, your temperature, and all this stuff, right? One thing that will occasionally spontaneously come about in water, again, we don't have to put any extra energy other than a little bit of heat into this to make the system to make it happen, right? Spontaneously, they form these bilayers. Can you see that if this was very, very large, you'd end up with this region in here? Is there going to be water in here? No, water's going to be excluded. These are all these hydrophobic, long fatty acid tails. And then there will be water inside and water outside. Okay? This is called a liposome. Liposomes these days are used for a lot of interesting biological techniques. Look at this little pocket inside that's formed. We're going to see some things where we can place a molecule of interest inside that liposome, inside that little ball, and have this fuse with the cell. And when it fuses, I'll get into this a little more later, it will insert what was inside the liposome into the inside of your cell. Okay, this gets really interesting. Okay, so this is a liposome. Notice, now how many, you see why I wanted to overly simplify this? Remember, carbon, 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 glycerol, phosphate, right? Here we go. One of those, another one of those, another one of those, so many of those. But we've only begun. When you get a lot of these, you end up with a phospholipid bilayer. Now again, you got to think 3-dimensionally. This is a little chunk of it, but it extends on a long way that way, a long way into the screen, a long way out of the screen, huge. If this were part of the cell, that cell would be bigger than the building being surrounded by these phospholipid bilayers. Okay? These phospholipid bilayers are a major part, well, they are the major component that make up the endoplasmic reticulum, the plasma membrane that surrounds and defines the whole cell, and the nucleus, and the peroxisomes, and all the other organelles. Okay? So this is why these lipids, these fats, are important to life in much more than simply energy storage. They actually are the main component of our cells, that define our cells, I should say. Okay? So again, we've got a hydrophilic head group, hydrophobic fatty acids, fatty acid tails in the middle. They're excluding water. If you make this big, so here's the thing about these phospholipid bilayers, and I've already hinted at it, saying what we can do with the liposome. There is nothing that is really attaching one of these phospholipids to the other than energetically favorable positioning relative to water. But these are not connected to each other. In fact, they can move relative to each other like this all the time. And they do. Okay? So in the dimension of this plane, they are fluid. Okay? These guys can move relative to each other in the plane of that membrane. But it is very energetically unfavorable for any one of them to flip, to change, because you can see that in order to do that, this hydrophilic phosphate head group would have to go through the hydrophobic region of the bilayer to be able to come back out the other side. This essentially never happens. Okay? Now, there are specific enzymes that can cause this to happen, called flipases and flopases. We're not going to get into those, okay? But in general, they stay in the plane that they started. Okay? Why does that matter so much if they're all the same? Well, they're really not all the same. I've given you a pretty simplified view of what they look like. Actually, many of them have slight modifications to that phosphate head group, and they've got names like single-myelin, phosphatidylcholine, phosphatidylserine, phosphatidylinositol. And if you think about this, this is another view of a phospholipid bilayer. Again, hydrophobic region, hydrophilic region. There's water outside the cell, right? Outside the cell, there's lots of water. And inside the cell, which is called the cytosol, there's also lots of water. The reason that these are shown the way they are, with phosphatidylcholine on the outside and single-myelin on the outside, with these guys on the inside, is because the cell actively uses those flipases and flopases I mentioned to set them up in this orientation. So these two layers of the phospholipid bilayer, two layers, right, are called the two leaflets of the bilayer. There's the inner leaflet, which would just be on the outside of the inside of the cell is in here, and the outside of the cell. This would be the outer leaflet. Okay? And the cell specifically uses these to set up this situation where you've got certain ones on the outside and other ones on the inside. As a matter of fact, one recent technique I saw for determining when a cell had died, I mean, at this level, it's hard to know when something's alive and when it's dead. It's just a bunch of molecules floating around each other. There's really no difference, right? And one definition I saw recently of a living cell is it's maintained this difference in the two leaflets. And once a cell dies, it stops doing the work necessary to keep this, and it starts to randomize which phospholipid goes which way. All right. All right, but this is only the beginning. Remember, these things are fluid in their own leaflet, right? They can move relative to each other in their own leaflet. But because they can move relative to each other, things can be inserted between them. In this case, what we've got inserted is, this is cholesterol. This is the molecular structure of cholesterol. And cholesterol has some important responsibilities for maintaining certain fluidity properties of cell membranes. You need cholesterol. You don't need it as much as it tends to be in the American diet. But you need cholesterol to maintain the certain fluidity of your membranes. Questions? I've talked a lot now. Does anybody come up with anything they want me to talk about more or less? All right, I'm going to keep going. Please feel free to interrupt me. I'm more than happy to stop and answer questions or anything. Okay? But if you can insert these cholesterol molecules, you can also insert proteins. And this is getting to be a more accurate representation of a plasma membrane surrounding a cell. Okay? You can insert proteins. You can insert, this is a long, one chain of a protein that is pretty long, and it's got this alpha helical region we'll talk about later that very nicely fits into the membrane. Guess what kind of molecules are right here? It's all one big long protein. What kind of region would this be in the protein? Would it be a hydrophilic? Hydrophobic region. A hydrophobic patch on that protein that very nicely sits in the hydrophobic region of the phospholipid bilayer. Okay? You've got outside, you can attach, these are some of the carbohydrates that I'm not going to talk about, and a lot of these are very specific to an individual, such that when they're the wrong ones, they induce an immune response. Right? They also have other properties that they give to cells. So we've got proteins, big globular proteins all bunched together that go through the membrane. So all of these phospholipids here, again, they are still free to move about relative to each other. What does that say about the protein? Is that protein stuck in place? No. We can also move it within the bilayer. Okay, because these phospholipids can move out of the way and allow this to be dragged through here. So this is called the fluid mosaic model of lipid membranes. It's mosaic because it's made up of a whole lot of pieces, and not all of them phospholipids. We've also got protein stuck in there. We've got carbohydrate groups stuck in there, lots of other stuff. And it's fluid because stuff can move around within the plane of that. Now, considering that this is a really hydrophobic region of this protein, it would actually take energy to pull it out of the protein. It doesn't want to move out of the protein. It likes having its hydrophobic region stuck in these tails, and it doesn't want that out here. So it's going to stay very nicely inserted in that membrane in just that way. So if we look at this, right, I turned this on its side because basically that is what is making up this whole membrane. You can imagine if this was the level we were looking at, the cell that this encompasses would be bigger than the building. Right, the scale of that would be huge. This is just a close-up of one little piece of this phospholipid membrane. Questions about that? So as engineers, you guys can think of all kinds of interesting things to study about this, right? How much force does it take to pull this guy through? How fluid is the membrane, right? When you get several cells stuck together, how much force would it take to pull those membranes apart? What if we have, remember that liposome? It can come in and fuse with this membrane and inject. There's just all kinds of interesting mechanical, dynamic things that are happening there. And I happen to know from that Gem 4 at MIT, people are doing some really interesting work on the mechanical properties of, for instance, red blood cells relative to sickle cell anemia, for instance. Those sickle cells have different mechanics, partially given by the fluidity of the phospholipid membrane, and partially due to a whole lot of proteins as well. Again, proteins make life interesting. All right. That was lipids and membranes. Moving on now to the next molecule I'm going to talk about, which is DNA. I should say nucleic acids in general because we're going to talk about both DNA and its relative RNA, ribonucleic acid. And why do we care so much about DNA specifically, but also RNA? Again, it's because the sequence of nucleotides in this ladder encodes a sequence of the building blocks of proteins. Okay? So again, proteins are what make life interesting. The DNA tells us how to build our proteins. That's why it's so interesting. When you hear somebody say they're a molecular biologist, the molecule that they mean by molecular, really, is DNA. Because it was just in 1957? 52, right? 52. When Watson and Crick, right, elucidated, showed the world, by kind of half stealing some data from another woman that we almost don't ever hear about, unfortunately. Rosalind Franklin was her name. They showed us this structure. And they showed us this double helix. And they realized immediately how this was used to do some important things for the cell, how this structure was important for the cell. I won't go into that in the story. But the point here is that we are going to be looking at how this sequence of nucleotides tells the cell how to build its proteins, which are the molecules that actually do all of the function and work in a cell. And this is a two-step process. The first step is called transcription. I hope none of you ever had anybody, as a punishment, give you a dictionary and say, transcribe this, pages 400 to 530 of this dictionary, right? I have heard of such things being done. When I'm asking you to transcribe something, what am I asking you to do? Rewrite it, copy it. Okay? The word scribe here, script, means writing. Okay? So we're going to move this writing to another thing. And so that's why it's called transcription, is because we are rewriting the DNA code into an RNA molecule. Into an RNA molecule. Again, I will go into the differences and similarities between DNA and RNA in just a minute, okay? This happens in the nucleus of eukaryotic cells. By the way, this entire lecture is strongly biased towards eukaryotic cells, okay? What do I mean by eukaryotic cells? We are eukaryotic organisms, okay? We have a nucleus in our cells where we keep our DNA. We have all these fancy organelles, like the endoplasmic reticulum and peroxisomes and all these other things, right? Bacteria are called prokaryotic cells. They don't have a nucleus. So for them, they also, and this is going to be another major theme for me for a little while, they also do transcription and translation in exactly the same way that your and my cells do, but they don't have a nucleus where they keep their DNA. Their DNA is just kind of out there in the cell. Actually, it's in a region called the nucleoid region, but basically it's not separated by a membrane from the rest of the cell. So in our cells, this occurs, transcription occurs in the nucleus of necessity because we have a nucleus where we keep our DNA. Okay, our DNA is stuck in that nucleus. When we go to make a copy of that DNA, it has to happen in the nucleus. And it does. Okay? It's carried out by a protein, a set of proteins called RNA polymerase, specifically RNA polymerase 2. Don't worry about the 2. RNA polymerase. Polymer. What's a polymer? I'm hoping everybody in the room knows what a polymer is. Yes? Okay, good. This molecule makes a polymer of RNA. You can see this is a polymer of DNA. This molecule makes a polymer of RNA based on this DNA sequence. Okay, that's why it's called RNA polymerase. That's the first step. Then, this copy that gets made by RNA polymerase leaves the nucleus. It goes out of the nucleus through pores in the nuclear membrane, the nuclear envelope. It goes out into the cytoplasm, where it finds a ribosome. Remember those little purple dots I wanted to point out early? Called the ribosome. Okay? The mRNA, the message, the copy, right? If you were being punished by having to rewrite a dictionary, stuff you rewrote, leaves the nucleus. It goes out to the cytoplasm to find a ribosome. And a ribosome is the place where we turn that mRNA message. By the way, M in mRNA stands for messenger RNA. Right? And that's where, in the ribosome, is where we use that message to tell us how to build the proteins. I promise I'll get into the details of this. Transfer RNAs, tRNAs, bring the amino acids to the growing polypeptide. Okay, what am I saying? Blah, 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 a lot of words. Right? The protein is a long chain built up of little subunits called amino acids. I'll demonstrate this more apparently tomorrow, as I'm getting close to time today. Okay? And these transferred tRNAs bring in those amino acids one at a time to build the proper sequence in the protein. Okay, so two-step process, transcription and translation. Here's a little pictogram of, we've got our genes, our DNA, stored here in the nucleus. This is clearly a eukaryotic cell, right? We do transcription to make this copy of some little piece of the DNA. That message, the mRNA, leaves the nucleus, comes out into the cytoplasm where it finds a ribosome, and the ribosome uses that message, which is a copy of the DNA, to tell how to build this protein out of amino acids. We're going to go through this step by step here a little bit. Jimmy, what do you think about time? Should I end on time today, or can I keep going for a little bit? It's up to you.
I think we show up to 11.
I can go till 11?
Yeah, we have the discussion time, and then if we need to break for 10 minutes.
Okay.
For the next,
all right.
Which is at 11.
Good. I think I remember the schedule saying that this was, till 10:30. Oh, it was 10:45, wasn't it? I thought it was till 10:30. Good. I'll keep going for at least a few more minutes.
The discussion time.
Good. Questions so far?
What's the question here? What's the final purpose of the protein was made? Was it the RNA and then RNA, or is it from DNA? What's the whole thing? Whole story about?
So the whole story is about making the proper protein.
Yeah, what's that for?
Okay.
What is a final product for?
Okay.
That's gross or?
The final product is, again, proteins make the cell interesting. If there was nothing to the cell except lipids, it would just be literally a round bag of water. But we know that cells take on all kinds of shapes. I mean, look at neurons. A neuron is a cell. Man, does it have a crazy shape, right? Red blood cells have a very specific shape, right? That donut thing. Cells can move. They're very dynamic, and we'll see some examples of that, I promise, tomorrow. All of this stuff. Actin, microtubules, motors that walk on actin microtubules, all of these things are proteins, right? Channels that let stuff into and out of the cell. The channel itself is made of protein. Proteins catalyze chemical reactions, breaking down things to gain energy, putting things together to store energy. All of these functions are carried out by proteins. Literally, our proteins are what make our bodies the way they are. They give us our characteristics. On the macro scale, they read out as blue eyes versus brown eyes. They read out as longer leg versus shorter leg. They read out as all these different things are given by proteins. The instructions for how to build proteins are the reason DNA is important. And this whole process builds all the proteins you have in your body. Did that answer your question?
Yeah, that means it keeps the lives going on.
Exactly, exactly. Proteins make life interesting. That's right, that's right. Good. Others? Excellent question. Okay, so this whole process, the purpose of it is to build correct proteins. It's kind of like the body is a big, diverse building. Okay? My muscle cells are different from my liver cells, and my liver cells are different from my neurons, and my neurons are different from my pancreatic cells, right? And my pancreatic cells are different from my... So we've got all these different types of cells. In my analogy here, which very strictly limited analogy, right? Our body is made up of a lot of different cells that are different from each other. Okay? In this analogy, every cell in our body has a filing cabinet in it called a nucleus. Okay? And inside the nucleus of every one of our cells is a blueprint for how to build the entire body. Okay, so there's a blueprint for the whole building in the nucleus of each and every cell. All right, so the genes in my liver cell, the DNA in my liver cell is the same as the genes in my muscle cell. And those are the same genes that are in the nucleus of my eye cell. And those are the same, I don't go on, I will belabor the point, right? We have the full set of genes, the full set of blueprints in every cell in our body, with a couple of exceptions. Red blood cells get rid of their nucleus. Red blood cells don't have DNA in them. The white blood cells do, but the red ones don't. That's a very unusual exception to the rule. Almost every cell in your body has a full set, not just one full set of blueprints, but actually 2 sets of blueprints for the entire body in the nucleus of every cell. You got one from your mom and one from your dad. Okay? In the nucleus of every cell. And these came together when your mother's egg was fertilized by your father's sperm. Egg and sperm each only have one blueprint. When they get together, now that new cell has 2 sets. One from mom, one from dad. Those then go on to read out to become you. Okay? A very long and involved process. All right. So that's what our nucleus is. It's like the filing cabinet where we're holding the genes, which are instructions for how to build the proteins. So I won't belabor this analogy at all. Okay? Let's get into how we actually read out these instructions. That analogy with the blueprints and all, it's quite interesting, but keep in mind that it breaks down fairly soon if you try and push that analogy too far. For instance, if you have an actual blueprint of a building, I assume none of you are architects, but if you have a blueprint of a building, you can use that as an instruction to build the building, right? But also, if you've got a building, you could meticulously go through and reconstitute the blueprints for the building, right? Not so with genes. Once they make that protein, you cannot use that protein to deduce what that genetic sequence was that produced it. And I'll tell you why after a little while. It doesn't go the other way like it does with blueprints. Okay. All right, so let's look closely at a comparison of these 2 nucleic acids that I wanted to talk about. Okay. The 2 nucleic acids that I want to talk about are DNA and RNA. So why am I showing this sequence of letters here? The reason is there are basically 4 nucleotides: adenine, thymine, guanine, and cytosine. I'm not going to labor the point of naming those 4 too much because we almost always just abbreviate them as A, G, T, and C. Okay? What that means is, I'm going to jump from up here to down here. Okay? I want you to notice that as we go through this double helix, what's highlighted here in this light blue little box that you can barely see, this is 1 nucleotide. 1 nucleotide. All right? And that blue box gets exploded down here. Okay? So this purple bit here, this phosphate, I told you we were going to see phosphates again, didn't I? Right? This phosphate here is this little purple blob here. That's a phosphate. Okay? This red group, this is actually a sugar called deoxyribose, deoxyribonucleic acid, right? Deoxyribose is this red blob here. Now for all 4 nucleotides, A's, G's, T's, and C's, those things, the phosphate and the sugar, are exactly the same. Okay? So they make up the backbone of this molecule. Phosphate, sugar, phosphate, sugar, phosphate, phosphate, sugar, phosphate, sugar. You can't really see the next one very well because it's in the plane of going into the screen. But phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Boring, right? Not a lot going on there, right? Phosphate, sugar, phosphate, sugar. In the middle of this ladder, if you want to make the ladder analogy, it's the rungs of the ladder, right? The 2 uprights of the ladder are phosphate, sugar, phosphate, sugar, phosphate, sugar. In the rungs, we have these 4 different little groups called adenine, thymine, cytosine, and guanine. They come in the middle here. So this thymine, yellow, is represented by this half of this rung of this ladder. Right? That's 1 nucleotide. What DNA is a string, a polymer of nucleotides. Okay? That's all I want to say about this for now. I'll get more into this in a little bit. All right? Notice DNA tends to be double-stranded. What do we mean by that? This is not just 1 strand, right? There's not just 1 backbone here. There are 2 backbones, right? 2 phosphate sugar strands strung together. Not only that, but this was one of the big things that Watson and Crick did, and maybe a few people a little before them, were talking about the idea that every time you have an A on one strand, you've got a T on the other strand. And every time you've got a C on this strand, you've got a G on this strand. Those nucleotides, A with T and C with G, always bind with each other in the middle. Okay? So every time you've got a T here, you know you've got an A here. Every time you've got a G here, you've got a C here. Every time you've got a C here, you've got a G here. And every time you've got an A here, you've got a T here. Okay? Then the lab twists up. Okay, DNA. Double-stranded, base pairing rules. We call those base pairing rules, right? Because these are phosphate sugar and a base. So these bases pair with each other in such a way that T always binds with A. We'll say that again and again and again and again. Okay, A with T, C with G. RNA looks pretty similar. Really similar, actually, considering how amazingly different it functions than DNA does. I think if I could go back to grad school and start again and I could study anything I wanted, clearly it would be engineering. But if I had to make a second choice, it would be RNA. Right? I would love to study RNA. It's an amazing molecule. It has a lot of function in the cell. We're only beginning to appreciate it. But RNA also has A's, adenines, guanines, and cytosines But notice, it does not have thymines. It's got a slightly different base called uracil. But it's really no big deal because the base pairing rules still apply. Here, we don't have any bases paired with any other bases. But we'll see in a minute, it's important that when a molecule of RNA does base pair with either one strand of a DNA molecule or with another RNA molecule, those base pairing rules still apply. A now goes with U, but G still goes with C. Okay? There's no thymine in RNA, and that's why they chose thymine to show this nucleotide of DNA, is because they wanted to be able to show that RNA has a slightly different look. This one goes carbon here, and 3 more. This, 3 H's. This one just goes carbon here, to hydrogen here, no more carbon. That's the big difference between thymine and uracil. Okay. But anyway, it's got A, G, C, and U in RNA. The other difference is we no longer have deoxyribose. We've now just got ribose as the sugar. That's why it's RNA, ribonucleic acid, instead of deoxyribo DNA. Because of this one extra oxygen coming off of that carbon. I guess maybe it took a little bit of chemical work to figure out these structures. Millions of man-hours, decades of work to come up with this stuff that I get to tell you about in 10 minutes here this morning. This is one thing I love about science. Huge years and years, decades of work by hundreds or thousands of people, and we get to give these wonderful little summaries that are getting longer and longer as we learn more and more. It's an amazing enterprise. And I'm sure it's the same in every single field. Okay, so those are the major structural differences between DNA and RNA. I've highlighted here that we've got there's T's here and U's there. All right, it's a big difference. Yes?
If it's such a small difference in structure, why is it just the T and the U that's different?
Fascinating question. I wish I knew the answer. That deserves a prompt, accurate, concise answer that I don't have. It ends up, what happens is DNA doesn't really ever, because it tends to be double-stranded, that's partially part of the reason, doesn't ever fold in on itself. DNA is an amazingly good molecule at storing this sequence of letters and copying and transmitting that sequence of letters. But what it's really bad at, for reasons that have to do with these two differences that, frankly, I don't have the expertise to really talk on at length, it's not good at folding up on itself to give it a special structure or shape that would give it a different function other than storing and transmitting information. RNA has this amazing property that it often takes on what I'll just define probably tomorrow as a tertiary structure, where it can have catalytic activity as well as information storage. I'm sorry I can't answer you any better than that, okay? Other questions? Yes?
The blueprint you showed earlier, or does the DNA sequence changes with time?
What time scale are you talking about?
Okay,
so,
some years.
So we all started out, this is an amazing question and it deserves a little bit of time, okay? His question was, does the DNA sequence change with time? Okay? It's an excellent question. Let me tell you, as a general rule of thumb, no. But in really interesting ways, yes, of course. Okay? The reason I say start out with the answer no is because you and I and everybody and every living organism started off as a single cell with exactly 2 copies of the DNA like I was talking about, right? When that cell goes to make itself into 2 cells, that genetic sequence, HETC, right, gets exactly copied so that each new cell has exactly the same genetic code as the other. right? When those two go to divide and make 4 cells, exact copies are made, right? If you go messing with that much, your developing embryo dies. Okay? Embryology is a very delicate process. The development of our bodies early, right? It's like laying the foundation of a building. If you make a mistake in laying the foundation, the whole building is going to crumble and not work, right? So you've got to copy that very precisely over and over and over again. So in that sense, now I've got some trillion-odd cells in my body now, right? And they are all exact duplicates of that original cell that was created when my father's sperm fertilized my mother's egg. So in that sense, you really hope no, right? That sequence is maintained for every one of those copies. In the evolutionary time scale, mutations do happen. I myself have a mutation in a really important enzyme that probably, if I lived 200 years ago, would have taken me out of the evolutionary race soon after birth, right? Fortunately, we live today with our modern understanding. I can be here in front of you today, right? But these mutations build up over time and give rise to all of the diversity of living things you see. All the diversity between bacteria and an oak tree and a snake and me happen because slowly changes in DNA built up over time in those lineages. So on the short time scale, no. That sequence stays the same in my whole body, right? In the big time scale, yes, and those changes are interesting.
Okay, so this means the DNA sequence doesn't contain any information regarding our memory?
Aha! No. So you mean our conscious memory given to us by our brains?
Yeah.
No. It codes for proteins that make cells take on interesting shapes like neurons, and those memories are encoded in the wiring of our neurons in ways that we don't know yet. I still think this is the one big brass ring out there in biology, is figuring out how memory is encoded. Once we figure that out, we're going to know a lot more. We don't know that yet. But it's encoded not in the genetics, but in the neural network itself, the network of cells. Now, did that get close to answer your real question then?
Yeah.
All right. Good. Great. Others? Yes?
What can make DNA code change?
There are several things that we come in contact with every day. Radiation from the sun. This is why, if you take your kids out, you want to put good sunscreen on them, because overexposure to the sun can build up mutations that can lead to skin cancer. Many chemicals, several chemicals in cigarette smoke can damage the DNA of the cells in your lungs, which gives rise to lung cancer. So you can see when mutations do happen in an individual, it's almost always, I would say 99.99999% of the time, detrimental. Okay? We don't want to mess with that too much. And many carcinogen, anything called a carcinogen, it means it's going to lead to cancer, happens because it mutates your DNA. Certainly anything called a mutagen, that's what it means, is it's going to mutate your DNA sequence. And so these chemical or radiological things can come in and break apart these bonds, and strange things can happen. And I'm not going to get too much into details of what tends to happen frequently, but yes, some of those things. Okay? This is why smoking is bad for you. Even if it does make you alert and happy. Yes?
Genetically modified crops, for example.
Yes.
What is the percentage of DNAs that they modify?
0.00005%. It's a very small amount of the total DNA. We share about half of our DNA with a banana. Okay? I'm not kidding, I'm not exaggerating. This is because so many of the things our cells need to do to live, that banana plants' cells need to do to live, right? And so to make changes that we want to see, like giving it a pesticide resistance or something like that, we only need to change 1 or 2 genes out of literally hundreds of millions of genes. Okay, so very small amount gets actually done. Great questions. Yes?
How successful for right now to transfer a gene or specific function to another cell to acquire that?
We're really good at that now. What's that? Was that your question?
Yes.
Yes. And of course, it depends on what type of animal you want, what type of organism you want to do. If you want to do a plant, people don't mind so much what you do to plants, right? So we've got things called gene guns that we can put. It's crazy easy to genetically modify a plant. I mean, crazy easy. I would have never believed it. You take little bitty nano-sized gold particles, coat them with the DNA of the gene you're interested in, literally shoot them into a plant cell with some very small percentage, but when you're dealing with hundreds of thousands of cells in a microscopic slide, it's no problem, right? That DNA is going to happen to come off that gold while it's in the nucleus of the cell. The cell then says, hey, there's a string of DNA in my nucleus, let's get it into the genome and express it. It's crazy how easy it is in some cases, right? Same with bacterial cells. We can transform them. I think you're going to transform bacterial cells in one of your labs. I know you're going to transform some cultured, I'm pretty sure human fibroblast cells in Joy and Manister's lab this week. So you're going to actually transform some cells, I believe, in the next 2 weeks. So it's terrifyingly and excitingly easy to do. It's both at the same time. And that was another point I want to come back to. We're going to transform that corn crop with genes that we take from an insect. When we make insulin these days for diabetics, we use it by expressing the protein insulin in bacterial cells. A human gene expressed in E. coli, in bacteria. My point here is DNA is DNA is DNA, and it doesn't matter whether it comes from a bacteria, or a daylily, or an oak tree, or a rabbit, or a cow, or meat. It's the same, and it works the same. We're really getting at the fundamentals of how life works. How does this all happen? Yes?
I have a question. You know when you said that you can't go backwards and move to normal, were you not taking into account like version transcriptase?
Yes, even though if you do use reverse transcriptase, she's asking about an enzyme that I'm pretty sure we originally found it by studying HIV, right, which has reverse transcriptase. And the idea is it starts out as RNA and goes backwards to make DNA, which then gets inserted in the host protein, host DNA. Reverse transcriptase, remember, the reason HIV is really hard to fight is because it's very mutagenic, right? It changes very rapidly. That reverse is very very low fidelity, right? It's copied back with a lot of mistakes. And if you go from protein, then it's even more, and I don't know of any situation in nature where we go from a protein sequence to an RNA sequence to a DNA sequence. But you're right that we can go from something of an RNA sequence to a DNA sequence, but it's very, very bad copying, large number of errors made in doing that. But you got me exactly very good point there. Did I answer you appropriately? Okay, good.
By changing the DNA sequence, can I somehow control the different attributes of any cell?
Absolutely, right? By changing the DNA sequence, you change what proteins that cell will make. And you can have it make a different protein than it ever made before. You can have it make a different version of a protein it made before. You absolutely can change the properties that that cell has. What's that?
For example, by changing the DNA sequence, I can control the refractory period of a neural cell, a neuron.
The what rate of a neuron?
Refractory period.
If you have a gene from one neuron, well, it's tough to get that specific yet. Okay? Probably yes. I haven't done it. I haven't tried it. But almost certainly yes. You could. Yeah, by giving it different voltage-gated ion channels that are more efficient or less efficient, you could do that, yes. Okay? Good. Excellent. Please, more questions. We're well into our discussion period time here, so yes.
Since DNA and RNA are so such similar, why is RNA
Great question. Since they're so similar, again, why does RNA typically act as a single strand? All right, when I say single strand, I'm kind of oversimplifying it a little bit. RNA does tend to act as a single strand, but really often that strand loops back on itself and makes those bonds between its own molecule. So you could consider this region double-stranded even though the whole molecule is only single-stranded. Okay, and that's going to be an important property when we look at tRNAs tomorrow. Okay. Yes? Okay, great question. Her question was, DNA gets maintained in the cells, RNA can disappear. Why? If anybody in the room has ever done any RNA work, you will be well familiar with the fact that our cells and even the surface of our skin all over the places has enzymes specifically designed to cut up RNA, to chop it up, to break it down into its nucleotide that they can be then reused to make a new RNA sequence, okay? So once it gets out into the cytoplasm, there are other proteins that can come along and protect it and allow it to be translated over and over and over again. But if the cell decides, I've got enough of that protein, it can seek out that specific RNA for that protein and chop it up so that it doesn't make any more of that protein. The cell has many ways of maintaining a balance. It wants the right amount of the right proteins, not too much, not too little, all the time, and it's constantly chopping up mRNAs to as one mechanism for regulating the amount of protein that gets made from that mRNA.
So that is the DNA stays in nucleus, so
Nicely protected in its nucleus, and it's well protected by a lot of other proteins that are in there as well. As a matter of fact, when we do have enzymes that are meant to cut up DNA, and we have something we're going to talk about tomorrow that are really important called restriction enzymes, those enzymes come from bacteria trying to kill competing bacteria. Okay? So they produce these things that chop up DNA as a way to kill off their competitors. And we said, hey, that's really interesting and useful. We're going to use that enzyme to help us do our biotechnology. I love it when humans do this. Find something that is meant to kill and learn from it and learn to use it to our benefit. And we do it pretty regularly. Not me, I'm not that smart. Can I see another hand? Yes?
In the previous slides, you mentioned about ribosome. Lipid by their
Ribosome, yes.
Lipid by their structure very similar to the cell membrane. So it can easily fuse with cell membrane.
They can easily fuse, not with the cell membrane, but with that membrane called the endoplasmic reticulum. Okay? I hope I didn't confuse you. Ribosomes really aren't made of lipids. They aren't similar to any lipid membrane, but they can very much embed themselves in endoplasmic reticulum. Right, ribosomes. What did I say?
He said
Oh, liposomes. Thank you. I apologize. Thank you. Okay, liposomes. They are surrounded. Okay, so. Wow. Now repeat your question, please. I apologize.
I'm curious about the liposome.
Yes.
Okay? This double bilayer lipid structure.
Yes.
Is similar to the composition of the cell membrane.
Yes.
I'm wondering if it can be used as the genetic or drug delivery carrier to transport something like that from outside into a cell. Can you give us some comments about that?
Yes. The answer to your question is absolutely. You got it exact. Right? Yes. So the cell has this normal function where it can take. Well, when it's inside a cell, we call it a vesicle, but it's basically a liposome, and have it fuse with the plasma membrane and expel its interior to the outside of the cell. We call that secretion. Okay? It's happening in a neuron. That can happen at the synapse, and that's how we get neurotransmitters from inside the neuron into that synapse to do the next one, right? You guys aren't neurobiologists, sorry. So, but the cell has many ways of doing that in reverse, right? Taking a liposome from inside the cell, fusing that with the membrane of that liposome with the plasma membrane and expelling stuff to the outside. We can use liposomes to do the opposite. Take this lipid bilayer, exactly as you said, with a gene we're interested in on the inside of that, and putting it under the right conditions so that it fuses with the plasma membrane and it inserts what was inside the liposome into the inside of the cell. And that does happen. Again, those lipids are exactly the same in both, right? The cell and the liposome. And as a matter of fact, the cell has specific proteins embedded in that lipid membrane to recognize those and say, hey, come here, fuse with me, and put your interior into the interior of the cell. Okay? As a matter of fact, that's one of 3 or 4 major ways we have of inserting genes we're interested in into new cells, liposomes. And I think Joanne might be having you use that technique. Liposuction is what it's called. It comes from transfection, which was what we used when we originally were putting new genes into a cell, transfecting them. It's called liposuction, using liposomes to put new genes into a cell. Did that answer your question, Mona? Okay, good, sure, yes.
So compared to translation, some of the translations and all that, is liposuction beneficial?
Liposuction is one of several methods of doing a transfection. There's also electroporation, where you just put the DNA in there and give the cells a little shock. And especially if they're E. coli cells, they're really good at taking in DNA at that point. You can also, and this is another thing we do quite frequently now, is use viruses that have had their disease-causing genes taken away, put our gene in that virus. The virus's whole job is to insert its DNA into the cell. So those are 3 of the major techniques we use. Heat shock is maybe the 4th. Any of those. You put the cell, especially E. coli cells, you put them under stress, and man, they look for any solution at all. They're taking in any DNA they find and trying to express it, trying to get themselves out of this stress. Okay? Yes?
I want to ask how to make myself. Just put the phospholipid into the water and then vortex, we can get the myself?
Yes. Yes. You might have to play a little bit with how much salt's in the water. You might have to play a little bit with the temperature of the water and the concentration of lipids you put in, but they spontaneously form under certain conditions. You don't need to put any extra energy.
And then how to transform myself to the new cell? From the single to the double layer.
Right. My understanding is it has strictly to do with the concentration of lipids. You up the concentration of lipids, they want to get more of them packed into the same place, and by doing so, they spontaneously form a bilayer. Yep. Now, there are companies who specialize in making these liposomes that we're going to be transfecting with that can tell you a whole lot more about that. But my understanding is it's almost strictly to do with the concentration of lipids in the water and the salt conditions in it. Yes. Maybe last question? Yes, good.
Basically, I think we can consider that as an invasion for cells.
Yes.
So how does the cell stand for those are foreign materials and initially
It's a great question. Her point is, we can think of this transfection as kind of an invasion of that host cell, and how does the host cell, how can we get the host cell to recognize that as an invasion and defend itself? Our hope with this is that the cell won't recognize that as an invasion, right? If we want to transfect that cell, we really want that cell to say, oh, this is just a DNA molecule that must have come away somehow, I want to use it, right? That's what we hope. And fortunately, if we keep things clean enough, that's pretty easy to do. DNA, again, DNA is DNA is DNA. And there's no way, if all you're using is DNA, there's no way for the cell to recognize foreign versus endogenous versus natural DNA. Now, when you start thinking of proteins and lipids that have all these identifiers on them, many, well, especially in our bodies, we have the immune system. At the cellular level, we might not be very good at protecting ourselves, right? Because your body doesn't care if it loses a few cells, it regenerates new cells all the time, right? You cut yourself, you make new cells, whatever. Your stomach lining is actually replaced, what, once a week? Other things are, every 7 years, almost every cell in your body gets replaced, right? So you're really a new you. But anyway, at the cellular level, we're not good at recognizing invaders. It's only at the tissue and organ level that the body starts to mount an immune response, including antibodies and other things that are specifically meant to recognize foreign invaders and say, hey, this guy doesn't want to be here, right? So at the cellular level, it's not very good. We're not, and we've used that to our benefit, right? To be able to transform, as you said, the crops to make them antibiotic resistant, pesticide resistant, and so forth. So it's not, the cells aren't that good at it. Bodies are pretty darn good at it. You are under constant attack by bacteria and fungi. We've all breathed fungal spores since we've been sitting here. If you didn't have a good immune response, you'd be dead like that. Not like that, but soon, right? Within a few days. So we're under constant attack, and our immune system is vigilant. But on the cellular level, not so much. Great bunch of questions. I'm looking very forward to continuing with you tomorrow. We'll get into transcription and translation and making proteins and how we're going tomorrow. Thank you, guys.
I told you Scott is going to teach you everything about cell biology. The good news is he's coming back tomorrow. Thank you, man. We're going to take a coffee break. Cookie, coffee, right? Cookie. Okay. Some food is outside. Let's come back at about 11:15. So it's that clock is not right. Oh, I was looking at that. So 11:20, 7 minutes. Meanwhile, if you want, for those of you who haven't got the T-shirt, you're welcome to pick up the T-shirt. What it might do, depends on how long you make this next one, right? If you make this long enough, it will be you don't want to make another but if you make 2 of these, they are large or extra. Now, this one, you would do very nicely where you have one hand here, you make another one here.
Also, if you just put a head here, that's going to try to take that head just the distance of the, right? It's only going to be halfway across this biway. See what I mean by that? This biway is like this. This is halfway, and this is halfway. If you only put a head here, you're going to have here and your other head here. This is not going to run insert. Right? The hydrophobic head will not want to sit here in this hydrophobic region. But if you make two of these, right, you take this and copy it, now what you got is, and then that again, and then that will insert very nicely in here and stay right in. Is that what you meant?
If I can create a longer tail.
Yes.
And then from head.
Yes.
So it's a new.
Then it would do
it's a new
if you made the tails twice as long and then a head,
yeah.
you're good to go. Yep, that's going to work good.
Okay.
There's water. Yep. So hydrophilic. Yep. And any protein that has a hydrophobic region that's inside the cell, it's going to fold that hydrophobic region to the inside to protect it from the water. Okay. Oh, great questions. Great questions.
This is very interesting. How the protein have the hydrophobic and the hydrophobic domain to penetrate.
We'll talk about it tomorrow. Yes, it is fascinating. I remember first being told about how proteins fold to give them their function. I didn't believe it. Even today, it absolutely astounds me that all you need is the right sequence of amino acids, and that protein will fold on itself correctly to give it its function. It's amazing to me. Okay, so some proteins happen to fold themselves in such a way. So think of DNA as a three-dimensional thing, right? It's got this double helix like a spiral staircase, right? Some proteins fold to where they have regions that fit beautifully into that DNA. Okay? So they match very nicely with that double helical structure. And then they have another region, that's called the DNA binding domain. Then they will have another part of the protein, same protein, right? Part of it inserts, and part of it has whatever function it does on the
This is called DNA binding protein. But the other, we can synthesize basic DNA sequence to recognize the antibody.
Yes. Similar. So what I'm not exactly sure everything we're talking about, but what we might do is we know the DNA sequence of the proteins that bind with DNA, right? So we can take that gene sequence, right? So DNA binding domain, right? And we can synthesize that binding domain with whatever other functional domain we want. Express that, and now you've got a protein that binds that DNA but has whatever function you've added to it. Recombinant proteins. Did I answer your question? Not really. I'm not sure what your question is. Ask again. Epidermal. Okay.
Yes.
Yes.
We can synthesize the spatial sequencing.
Yes.
The DNA can be folded.
The sequence, right? The differences between those four bases is too small. Antibodies cannot recognize the difference. Okay? Now there are some DNA binding domains that can recognize specific sequences. Right? Antibodies don't do that. Okay? We might be able to engineer antibodies that do that. I've not heard of that. Okay? And there are other enzymes, like these restriction enzymes that I hope I get to tomorrow, that do recognize specific DNA sequences and cut DNA at those specific sequences. We use this in biotechnology all the time to help us clone and splice genes into
a big crowd for this.
So we find specific sequences and cut them at those, but not antibodies.
No, I guess it just can't.
Antibodies basically bind something and then say, hey, white blood cell, come here and kill this thing. That's their job. So we also use the engineers. That's their job. The DNA may not be specific because everyone isn't has to. That is good. Excellent. To hear more. I want to tell more. Yeah, okay, good. I will see you tomorrow. I can't make it happen before then.
During the lecture, it was tough to try to think of, well, who are they going to know? Yeah, and I really
I'm excited to hear it. Tomorrow too.
been doing, students.
I hope so too.
Here we're going through basics. I like more basics things.
Well, I think that, we'll do it. But if there's questions, a lot of people, nothing, any aspect of discuss it yourself. It's not like
I think to the registration desk.
Different size. Different size.
Forms.
I'm waiting. Question for Greg. Yeah, but
Okay, it's my great pleasure to introduce you, Professor Brian Cunningham. Brian is in the Department of Electrical and Computer Engineering at the University of Illinois here. He's been doing a lot of interesting work in the biosensing area. So let's welcome Brian.
Well, for those of you that are visiting, let me add my welcome to you to University of Illinois, to Champaign, Illinois. Hopefully you get a chance not only to enjoy the laboratory, but also our terrific summer weather right here. What I hope to do in the next hour is to give some background information in the area of fluorescence-based biochemical assays, describe what fluorescence is, what fluorescence is used for, and then at the end describe how we're doing some work with special optical structures called photonic crystals that allow us to amplify or increase the sensitivity of any biological assay that uses fluorescence. So fluorescence is the predominant way of doing biodetection in life science research and in drug discovery research and in diagnostic tests for diseases, measuring things in blood and so on. So even though there are many different types of biosensors, some of which you'll learn about in the Summer Institute this week, fluorescence detection is by far more sensitive by about 3 orders of magnitude. And so fluorescence is really the method that is used predominantly for detecting things at really low concentrations. So one application for fluorescence is shown here. Is fluorescence allows people to visualize living things such as cells or bacteria. I'll talk a little bit more about this image later, but this is a multicellular organism where different parts of the organism are lit up by different fluorescent chemical dyes. Another big application for fluorescence detection is in the area of DNA gene expression detection and DNA sequencing. But what I'm showing you here is a fluorescence image of one small section of something called a DNA microarray. So in the talk I'll describe more what a DNA microarray is, but what I'll say now is that this is the method that's used for telling what genes are turned on or turned off in different kinds of tissue, and being able to tell the difference between what genes are turned on or turned off, for example, between cancerous and healthy tissue. So in this picture, every spot represents a place where one biological assay is being performed, where each spot contains one small sequence of DNA. And so a single DNA microarray will actually contain millions of individual spots, allowing researchers to look at millions of different genes and whether they're turned on or turned off all at the same time. Another application for fluorescence is detecting proteins that are in the body. One big example is something called detecting things called biomarkers. So if a person has a tumor somewhere in their body, the tumor can be very small. You may not be able to feel it, may not even show up on an X-ray yet, but those cancerous cells will produce characteristic proteins that will come out of the cell and end up circulating throughout the blood everywhere in the body. And so the trouble is, the tumor may be very small, and those proteins will be diluted throughout the body, throughout your entire blood supply. So the concentration of those proteins will be very, very low. If you were just to take a couple of milliliters of blood out of your finger, you may only have a couple of individual proteins that would indicate the presence of the cancerous tumor somewhere inside the body. One of the technologies that my group and other groups work on are things called protein microarrays, similar to the DNA microarrays, but fluorescence detection is used where each of these spots represents a place where an antibody that allows you to selectively detect a single individual protein that is expressed by a tumor on this location on a detection device. Again, I'll describe more about how this works later, but this is a technology that's used for detecting not only presence of cancer, but also protein biomarkers that are involved in Alzheimer's disease, Parkinson's disease, cardiac disease, and so on. So in my talk today, what I'd like to do is to describe how fluorescence works, then talk about some applications in a little more detail, and then talk about some of the latest research. So I'll start off by talking about exactly what fluorescence is. And to consider that, first let's think about what a photon is and what a wavelength is. So again, not knowing the exact, not everyone will have the same background in this group. I wanted to go back to basics a little bit. So we know, I hope, that light has a particle-like nature. In other words, it's kind of like photons. Individual photons of light can be used to excite electrons, for example. But light also propagates as a wave, and so it has a wave-like nature. Now, what I've shown here is just a cartoon representation of a red wavelength of light with a wavelength of 650 nanometers compared to a blue wavelength of light, where a blue light has a shorter wavelength, around 488 nanometers. Now, an important thing to note here is that the energy of an individual photon of a particular wavelength, the relationship between energy and wavelength is inverse. Oh, the laser died. It didn't last long. Oh, here it is back again. So, energy and the wavelength are inversely proportional. So energy is equal to Planck's constant multiplied by the speed of light divided by the wavelength. So all that means is that higher energy photons have shorter wavelength. Okay, so now let's think about what happens or how wavelength of light interacts with molecules. So here I've just drawn a simple molecule, the water molecule. We know that the water is comprised of the nuclei, the atomic nuclei with protons and neutrons shown here. But also, the molecule is comprised of plenty of electrons. So as we know, these electrons are somewhat free to move inside the molecule. It's kind of hard to strip them away. Water doesn't like to totally give up its electrons. But electrons have negative charge. So if we go back and think about a wavelength of light, what I've represented here is not only the wavelength, but also the fact that this light propagates as an electromagnetic wave, which means that it has an electric field that oscillates between positive and negative, and very, very rapidly as the wave propagates through a molecule. So if we consider the effect that an electric field will have on negatively charged objects, if we remember Coulomb's law, tells us that an electric field exerts a force on charged objects. These electrons will experience a force from the electric field from light that propagates through them. So we can kind of think of the molecule as being subjected to a battery that we can, at any one instant in time, the battery will apply an electric field with one orientation. But because we have an electromagnetic wave, we can think of the battery as going between positive and negative polarity over and over again very rapidly. So it's almost as if we took our battery and just rotated it around like this. And so within one wavelength of an electric field, the electrons in that molecule will see positive charge or positive electric field sometimes, negative other times, and then oscillating back and forth between positive and negative. So then if we were to see what happens to the electrons in our molecule, they'd be literally bouncing up and down, oscillating inside the molecule. And so in this sense, a molecule is a little bit like an antenna, where if we have our cell phones have an antenna in them, the electric field, radio wave, oscillates electrons, causes the antenna to pick up the signal. In the same exact way, a molecule is like a tiny antenna where the electrons undergo oscillation in response to applied electric fields that oscillate from an electromagnetic wave. So just to give you an idea of the scale, it's kind of like our molecules are like awash in a sea of giant electromagnetic waves. Just to give you a sense of the scale, a water molecule is the size of about 0.1 nanometers, protein molecule, typical one, around 20 nanometers. A wavelength of red light, 633 nanometers, is of course much bigger. So you can imagine if you're sitting here, if you're an electron in this molecule, it's like these waves are passing over you, moving your electrons up and down very, very rapidly. So what does this have to do with fluorescence? So fluorescent molecules are more complex than water molecules, but I've shown a representative one here. This is a fluorophore called fluorescein. But you can see that it's comprised of carbon rings, some single and some double bonds. Has some hydrogen and oxygen in it. But just like water molecule, this is comprised of atomic nuclei grouped up together that also has electrons in it that are semi-free to move back and forth. So what happens when we illuminate this fluorescent molecule with some light? Well, I wanted to give you the electron picture because what's happening physically is that the electrons are being oscillated inside this molecule. However, the electrons, as they oscillate, will only occupy discrete energy levels, kind of like a semiconductor like silicon. So they may have a preferred ground state, their lowest energy level, but there will be gaps between the ground state and the next excited state and the next excited state after that. The same way as electrons can occupy discrete electron orbitals. Same thing happens even in a larger molecule. And so in this diagram, again, we see that there's gaps between states, but that as we go up from the ground state, we have higher and higher levels of energy. So let's think a little bit about photons and how we might define how they interact with molecules. I just wanted to lay down a little bit of terminology here. Because sometimes these terms are used incorrectly or substituted for each other not in the right way. So we can define first a photon flux. Mathematically, photon flux is just a number of photons per second that are flowing through a particular area. And so photon flux, phi here would be photons per second per unit area. Second thing that we can define, if we wanted, we know that now all photons are not created equal. Some photons have higher energy than others. And so we need to account for that from the fact that photon flux is not enough to tell us how much power that is going into some molecules that we're illuminating with light. So to define that, we can define something called photon power density, which is just the photon flux multiplied by the energy, hc over lambda, of the photons coming in. And so for this, we'll just assume that we're using a laser, one wavelength. All the photons have the same wavelength, and so we can define this photon power density as units like watts per meter squared. So like power density. Now, next thing we want to define, we'll see that it's important to think about not just all photons, but photons that are defined in specific bands of wavelengths. So we'll see that we may not have just one wavelength, we may not want all wavelengths, but we might want to consider all the power that's in just one specific narrow band of wavelengths. And so that's defined something called photon irradiance. This is the flux divided by the band of wavelengths that we're going to consider. And then finally, what we call intensity. So we normally think of it as intensity as kind of a combination of the photon irradiance and the photon power density. So it's the energy versus time for a defined range of wavelengths and collection area. So it's this EP, it's our photon irradiance multiplied by the energy of the individual photons coming in. Okay, so now let's think about the energy band diagram of a fluorescent dye molecule and what happens when that fluorescent dye molecule interacts with light. So what I'm showing you here is something famously called a Jablonski diagram. Jablonski diagram is just an energy band diagram for a molecule where we have our ground state and our excited states. Now, if we want a molecule to absorb a photon of light, that incoming photon has to have at least enough energy to excite an electron from the ground state to above the first excited state. So greater than or equal to this gap in energy is required to excite that electron to a higher energy level. So once an electron is at this higher energy level, I mean physically it's wiggling back and forth, but schematically on this Jablonski diagram it's sitting in a slightly higher energy state. It wants to get back down to the ground state. Things always want to get back to their lowest possible energy level. So how can it do that? Well, for a fluorescent molecule, it can give up some of its energy by emitting a photon. Giving up a photon that goes out into free space that has an energy represented here and a wavelength as the gap between the first excited state and the ground state. Now we notice here that we might have a little bit of extra energy left over. And so what happens to that? So this extra energy might be transferred into the molecule through mechanical vibration, where it may, for example, absorb it in the form of heat that it has to give up some other way that does not involve giving up a photon. So what you notice here, the most important thing about a Jablonski diagram is that you can see very clearly that the energy of a photon coming in, exciting the molecule, is bigger than the energy coming out. And so this difference between energy going in and the energy coming out is what's called the Stokes shift. I'll talk about that in just a second. Unfortunately for us, giving up a photon that's fluorescently emitted by a molecule is not the only way that an electron can get back down to the ground state. So we prefer that electrons get back down to the ground state by this fluorescence process. Each time that happens, we get one photon coming out. However, that's not the only way. So for example, if an electron is wiggling inside the molecule and is unfortunate enough to slam into one of the atomic nuclei, I mean, they occupy physical space, and so it's possible that the electrons can do that. In that case, a photon won't be given up, but instead, the electron will get back down to the ground state by giving up its energy in a collision where we have something called an intersystem crossing, or an IC process, that results in just transfer of mechanical momentum from the electron to the molecule. It's also possible, since our fluorescent molecules don't usually exist in a vacuum all alone, they're usually surrounded by many other molecules. It's possible that that electron that's excited may give up its energy mechanically to a neighboring molecule. This is called an intersystem crossing, or an ISC. So this little gap here represents energy getting sucked away by a nearby molecule. And then that molecule, actually, it may itself fluoresce at a different wavelength, or it may itself give off its energy to an intersystem crossing. So all these other mechanisms that are possible kind of compete with the process of fluorescence, our desired process. The difference, I guess if you remember nothing else from this lecture and want to know something about fluorescence, remember this. The difference between the energy that's absorbed and the energy that's emitted is called the Stokes shift. And so the Stokes shift is very important because it allows us to differentiate photons that come in from the photons that come out. So for example, if we're trying to excite fluorescence in a molecule, we may be using a very high intensity laser source, blasting our sample with a very bright laser, so we'd have a lot of photons coming in. However, because of all these other processes that compete with fluorescence, the number of photons that come out may be substantially smaller, maybe even orders of magnitude smaller. But it's our goal to detect these emitted photons in the presence of all these laser photons coming in. And so because they have different wavelengths, as we'll see, it'll be possible to use an optical filter that will allow you to block these photons from your detector and to see only these. So that only happens because of the Stokes shift. Okay, so now these processes compete with each other. They can be defined mathematically by the rates at which they happen. So if you were able to somehow like sit down on a fluorescent molecule and to sit there with your stopwatch and then count the number of photons that get back down to the ground state by each of these different methods, you could define what's known as a rate, kind of defined as the number of events per second for each of these processes. So what you would hope is that your fluorescence process would have the fastest rate compared to all these other things. So in other words, if we excite a whole batch of electrons up to this higher level, we want them all to get down to the ground state through this fluorescence process and to ignore these other ones. So as long as this one happens much more quickly, we can have a much more efficient fluorescence process. So the way that this is defined mathematically, or the way that fluorescent molecules are compared to each other for their brightness or goodness, is by a number called the quantum yield. So the quantum yield is simply defined as the number of photons that are emitted by fluorescence divided by the number of photons that are absorbed. And so you can define the quantum yield by comparing the rates of the fluorescence process and all these competing processes. So we can define the rate of fluorescence divided by the rate of fluorescence plus the rate of all those non-fluorescent processes. If we divide those, we would get the quantum yield. And so for some particular fluorescent dye molecules, the quantum yield can be as high as 95 or 99%. For less good molecules, can be as low as 10% or 20%. But a quantum yield also is not constant, unfortunately. And so what we'll see in a minute is that fluorescent dye molecules that can start off having a terrific quantum efficiency or quantum yield can degrade over time, get worse and worse the more you use them. So another definition that you see with fluorescence very often is something called the fluorescence lifetime. So the lifetime is mathematically defined as 1 over the rate. And so we have units where the rate has units of events per second. The lifetime has units of seconds. And so what the lifetime it tells us is that is something about the amount of time that electrons stay in that excited state. And so if we illuminate a fluorescent molecule with a photon, photon comes in and excites an electron to the excited state. It's not immediate that those electrons get back down to the ground state, but rather they exist in that excited state for a short, but very finite and measurable amount of time, usually measured on the order of nanoseconds. And so if we were to take a jar full of fluorescent molecules and come in with a pulse of laser light and excite a bunch of those fluorescent molecules up to their excited state at time equals 0, and then measure the fluorescent photons that come out as a function of time after that, what we would see is that we would have immediately a very high intensity, but then the intensity would die off, shown here exponentially, looks kind of gradually, but in fact this defines the lifetime. This decay would happen on the order of between 1 and 10 nanoseconds. And so what we see here is that the photons, or the electrons, decay back down to their ground state, not all in lockstep. They don't do it like they don't sit there and look at their watches and wait for 10 nanoseconds to go by and then all jump back down to the ground state. But in fact, it's more of a probability that they'll relax down to the ground state over a particular period of time. And so what we see is that if you were to measure lots of photons, some of them would decay back to the ground state right away. Other ones might happen a little bit later. But if we were to measure like the 1 over E magnitude of this decay curve, we would be able to measure what the fluorescence lifetime is. And so, in fact, it's possible for people to measure these very accurately and to know very precisely what the lifetimes are for fluorescent and non-fluorescent processes for particular molecules. Okay, so now let's think about that fluorescent molecule fluorescein a little bit more. And so what I'm shown here are two very important curves for the fluorescent dye fluorescein. So what's shown here is on a wavelength scale. So we have a low wavelength going to high wavelength, kind of going from ultraviolet wavelengths out to around like red wavelength out here. The curve on the left are the wavelengths that are absorbed by the fluorescein molecule. So what you see is that there's a specific band of wavelengths that are absorbed by the molecule. Not just like one precise wavelength, but actually over a small range of wavelengths. And so that happens because the energy levels between the ground state and the excited state are not totally precisely defined. Remember on my Jablonski diagram, I showed there were some dashed lines kind of representing different electron spin energy levels that are possible for electrons. And so because of that very small range of energies, even within a single energy state, we end up with a narrow band of wavelengths that are strongly absorbed. Now also notice that the curve on the right is the range of wavelengths that are emitted by fluorescein. Again, it's a narrow band of wavelengths, not just a single wavelength, but also notice that the gap between these two means that they have a Stokes shift. That there's a difference between the wavelengths that are absorbed and the wavelengths that are emitted. And so if I wanted to excite fluorescein molecules to give off fluorescence, all I have to do is to pick some light source that has photons that are in this energy band and inside the curve that's on the left. So for example, a common laser, question?
Yeah, which curve is which?
Oh, sorry, I should have labeled them. The one on the left is the absorption, so lower wavelength, higher energy. The one on the right is the emission. Oh, sure. A commonly used laser, or a common laser wavelength is 488 nanometers. That's an argon ion laser. If we happen to use that laser to zap some fluorescein molecules, it would be maybe not exactly on the maximum absorption, but somewhere on a pretty good spot on the curve here, and that would be enough energy to excite the electrons in fluorescein to the higher energy level. Just as an example, here's another fluorescent molecule. This one's called cyanine-5, or Cy5 for short. In fact, there are dozens, if not hundreds, of different fluorescent molecules that exist that are engineered by chemists to have particular properties. And so this is one such molecule that's been engineered to have absorption at around a red wavelength of 633 nanometers. So in the blue curve is the absorption, a red curve is the emission. So once again, you can see the Stokes shift between absorption and emission in the narrow band of wavelengths that are absorbed and emitted by Psi-5. But notice here that Psi-5, again, is comprised of carbon, organic molecule, has lots of carbon rings and carbon bonds in them. This one, as compared to fluorescein, is like a long and thin molecule. Oh, laser's semi-working. Okay, so it has a long and thin molecule rather than kind of a more bunched up globular molecule. So what we'll see as we go forward is that Psi-5 happens to be one of the fluorescent dye molecules that's used most commonly for DNA analysis and for protein biomarker detection. And so, this is an important one for my research group to work on. And so we talked about fluorophores so far. But, a question.
In the previous one, why is there kind of a double bump in a double peak in the absorption curve for Psi-5? Is that got any significance?
Yeah, so that's a good question. So, so here we see that the spectrum is not totally one peak, but there seems to be like a secondary peak here. And so there can be more, I guess, complex set of energy bands than what I've shown in the Jablonski diagram. And so, in fact, for this molecule, there can be like bands at even higher energy that can, so like a second higher energy level that can also absorb a photon. Okay, see what I mean? So this might be like the first excited state. This might represent the second excited state.
So does the amplitude normalize the energy of the radiation the same?
Okay, yeah, so here, the absorption and emission curves they're just normalized to have the same magnitude so you can compare them. But if this was a molecule that had 100% quantum yield and you could capture every photon that came out, then they might look the same. But it's never the case, actually. So the fluorescent molecule isn't smart enough to know what direction to shoot its photon out. And so if I have a fluorophore sitting on this table and I excite it, I mean, its photons will go everywhere in circle coordinates. And so if we're sitting up here with a microscope or a photodiode or something, we'll only capture a small fraction of them. And so without normalizing them, actually, the red curve would be way lower. Okay, so we talked about some fluorescent molecules so far, but what we're really interested in is using fluorescence to detect biological objects like DNA molecules, proteins, and cells. And so what I'd like to get the idea here is that in order to see biological molecules, the things like an antibody molecule, for example, antibody molecules, I guess in the biological world, are kind of big. So they might, antibody might have a molecular weight of 150,000 Daltons, meaning about 150,000 hydrogen atoms worth of mass. But still, even in the protein world, that's kind of a big protein. They are very, very difficult to see under an ordinary microscope. They don't have enough like height or optical contrast to see them very easily. And so the fluorophores are used or attached to molecules we want to be able to see so that we can visualize the molecule. What we're actually doing is visualizing the fluorophores that are attached to the molecule. So what's important is being able to attach fluorescent molecules to bigger molecules like proteins using things like covalent bonds that allow them to be attached very securely, not just rinsed away in solution. And so the way that that's done is typically to have a fluorescent molecule, like the ones I showed you, that has part of the molecule is engineered to give off light by fluorescence. One part of the molecule is engineered to make a covalent bond with specific places on protein molecules and DNA molecules. And so I'll talk about that more in just a second. But the basic idea is that you take your fluorescent dye molecule with the optically active part and the chemically active part, mix them together with your physically, I put them into a test tube together with the molecules that you're trying to label, and then you allow the fluorescent molecule to make attachments, covalent bond attachments to the molecule you're trying to observe. And so what happens is that it's not really possible to specifically control where the fluorescent molecule goes. And in fact, there will be like several places on a given molecule that a fluorescent dye molecule will attach. So a single antibody molecule may have not just one, but several fluorescent dye molecules attached to it. So how does that attachment happen? Well, here, again, for the chemists in the group, I represented an entire protein molecule existing here, where R represents whole big part of the molecule, where some parts of a protein have NH2 amine functional groups that are displayed on the outside surface. This happens because proteins are made out of amino acids. Some amino acids, when the proteins fold, are hydrophilic, so like to be on the outside part of the protein. And then a few specific amino acids have an NH2 functional group as part of them. And so very often NH2s find their way to the outside parts of proteins. And so any protein that has like a lysine amino acid group is going to have like a corresponding NH2 functional group on the outside part of the molecule. Now the fluorescent dye molecule, here R prime represents like the whole big section of the fluorescent dye molecule, but then the chemically reactive end is represented here. So this NH2 functional group would just love to make a covalent bond attachment with this COOH, called an aldehyde functional group, on the fluorescent molecule, and then kick off this part here, called an NHS group, as a chemical byproduct. So when that happens, again, just by mixing the two things together through magic of chemistry, the unwanted part goes away, but then we have our R prime, our fluorescent dye molecule, attached with a nice covalent bond to our protein molecule. Okay, so I'll just spend a couple minutes talking about problems with fluorescent dye molecules. Fluorescence, again, it is like a very predominantly used method for visualizing and detecting like presence of protein molecules and DNA molecules. But fluorescence suffers from like a lack of quantitation, meaning that a fluorescent dye molecule doesn't stay constant as a function of time. So again, if we think of our fluorescent molecule, we go back to think of this one here, and we imagine that this molecule is excited by a photon. So we have electrons wiggling rapidly, very quickly inside this molecule. When the molecule is in an excited state, the oxygen functional groups, like oxygen groups that are here and here, become slightly more chemically reactive than they would otherwise be. And so when the fluorescent molecule is in this excited state, and if it chemically reacts with some other nearby molecule through interacting usually with oxygens, the fluorescent molecule goes dark. It may no longer have the ability to have a very high quantum efficiency anymore. And so the more a molecule is in an excited state, the more you illuminate it, the more likely it is that it's going to chemically react and go dark on you. And so this phenomena is called photobleaching. Photobleaching basically means that the more you illuminate a fluorescent molecule, the dimmer it gets. And so if you were using fluorescent dyes to take images of cancer cells under a fluorescent microscope, the second you turn on the fluorescent microscope, start illuminating your cell, looking at it, it'll be nice and bright. But 30 seconds later, and you may not be able to even see it anymore. So photobleaching can happen very rapidly. And so you'll see that people working with fluorescent dye molecules to label things like proteins or cells usually will work with their solutions and bottles wrapped in aluminum foil, or do their work in a room that's dark in order to prevent even like the room lights or sunlight from the window from photobleaching their fluorescent dye molecules. So what that means is that if you're trying to measure fluorescence quantitatively, saying like the amount of intensity I get out is going to have some some meaningful value. Because of photo bleaching, the data that I take today might not give me the same number that I take 30 seconds from now. It certainly isn't going to match up with the data that someone else takes in a lab halfway around the world, where maybe they weren't as careful as you were with protecting their molecules from the light and so on. So photo bleaching is a big problem for quantitation with fluorescent dyes. Another problem with fluorescent dyes is a phenomenon called quenching. So with quenching, basically quenching means that there can be processes other than fluorescence where electrons give their energy to nearby molecules and that those molecules don't fluoresce themselves. So for example, we might think that if we want to have a molecule fluoresce very brightly, if we want to label a protein molecule fluorescently so we can see it really, really well, it might seem logical that more fluorescent dye molecules would be better. But because of quenching, that's not true. So what's shown here is the brightness is measured by a microscope versus the number of dye molecules per antibody. So what you see here is that at first, as you have one dye molecule, 2, 3, or 4, you get more and more intensity out. But then after a time, we start to have leveling out and then even decrease of fluorescence output. And so that's because as more fluorescent molecules are crowded together, the photon from one molecule that's emitted can be robbed by a nearby fluorophore, and then that fluorophore may not emit as a fluorescent photon coming out. And so actually there's a range of maybe only 3 or 4 dye molecules per antibody that's really optimal for seeing fluorescence output. What I'd like to do next now is talk about fluorescence instrumentation. What things do we use in the lab to see fluorescence? Well, basically, all fluorescence detection systems have all the things that are summarized here. We have some kind of light source that may be emitting lots of different wavelengths of light. We may have some kind of filter that's used to remove some of those wavelengths of light, so only a narrow band of wavelengths hits our sample where we're trying to measure. So our sample is going to fluoresce. So the fluorescent photons represented in red, actually they can go in all directions like I mentioned, but some of them might, by chance, make their way down towards a detector, a photodiode or a microscope imaging objective or something like that, that allows us to see them. So if we're illuminating with these blue high energy photons, they may be scattered all over the place. We don't want to detect them, so we need some kind of filter to remove those high energy photons from the optical path here before we can detect them down here with our emission detector. And so this second wavelength filter is called an emission filter. So there's several different light sources that are used in fluorescence. One particular one is just a tungsten lamp, but it's just like a microscope lamp. You might even have a tungsten lamp on your desk. So what do you know about a tungsten lamp? Well, they give off a lot of wavelengths of light in a smooth wavelength distribution like this, where some of the photons have high energy, but then there's a lot of photons coming out that have low energy, but are in the infrared, so not visible. So we know this because these light sources are very hot. If we touch them, physically, it will burn our fingers. And so tungsten lamps aren't really so terrific for fluorescence unless we can somehow filter out all these infrared photons. We don't want to necessarily cook our sample. So what can be used instead is a xenon lamp. A xenon lamp is sometimes called like a daylight lamp or a white light lamp. It's the same kind of lamp that's used in movie projectors, like IMAX theaters have huge xenon lamps. They also get very hot. So we see here like the wavelengths that come out kind of span from ultraviolet all the way out to infrared. A lot of infrared energy coming out here. So these have to be cooled. Again, we don't want this heat cooking our biological sample, will denature our proteins or kill our cells and so on. And so what's typically used in fluorescence is to take a little bit of mercury and introduce it into this xenon lamp. A xenon lamp works by having a small ampule of glass. It has 2 electrodes between which we apply a very high voltage. perhaps 2,000 to 10,000 volts, so that we get a a plasma or an arc between a tiny point, like a cathode and an anode. So within this tiny point, so it's like a point source of light, the gas inside the ampule is ionized and relaxes back down to its ground state by giving off lots of photons of different wavelengths. Now, if we add a little bit of mercury into the ampule, we would get a photon output characteristic that's more like this. So then the mercury is excited to a high energy state. The mercury then starts to dominate the spectral output of the xenon. And so after a short warm-up time, after the mercury evaporates inside the ampule, you see an output spectrum like this. So now what we see is that a lot of that infrared energy is gone, but we have specific wavelengths that are emitted in narrow band of wavelengths by a mercury xenon lamp. So this is important because people had to at first engineer their fluorescent dye molecules so their absorption matched specific wavelength bands of a mercury xenon lamp, so they'd be absorbed and they could excite their molecules. So if you had some fluorescent dye molecule that absorbed light here, you illuminate with a mercury xenon lamp, nothing would happen. And you just, since this is a fundamental characteristic of mercury atoms, you can't really adjust the wavelength. So it wasn't really until lasers came along that there was a lot more flexibility in designing fluorescent dye molecules for a lot of different applications. And so the good thing about a laser is that it's a very high intensity light source, one specific wavelength, and no other wavelengths at all. And so lasers are a very popular light source used in fluorescence. And so what's shown here are just a few common laser lines. 457, 488 I mentioned, 632 nanometers is a helium neon red laser, very commonly used. And so when you pick your fluorescent dye molecule, you're kind of picking the wavelength of the laser that you're going to use, but at the same time. Okay, so if we consider now like what's inside a fluorescent microscope, it's a little bit more complicated than the first diagram I showed you, but mostly if we take into account things have to do with the light source and the emission. So if we have a mercury arc lamp giving off a lot of wavelengths, including some infrared and heat wavelengths, first we have some filter, like a heat filter to block those infrared wavelengths, so we don't want the heat coming and cooking our sample. We might have a couple of different optical filters that would chop out some of those other unwanted wavelengths of light, so we have only specific band of wavelengths of light coming through. So we illuminate our sample from the bottom, and then using a microscope objective, we'd be looking down at our sample, gathering the fluorescent light that comes out. And so then using an excitation filter, called a barrier filter here, to allow us to see only the fluorescent emitted photons and not any other photons that were used for excitation. Okay, so this is what's used, for example, to take this image of a bacteria that I showed you up at the beginning. This is kind of an interesting one because not just one fluorescent dye was used, but 3 different fluorescent dyes were used all in combination. Each of the fluorescent dyes were chosen to permeate specific parts of this bacteria. And so we can see that this bacteria has compartments in it, some of which, so we have a stain that's a red stain. It's called a viability stain. That this particular stain can get into like the bacteria membrane. There's other stains that can permeate only dead parts of a cell, other parts that will only stain live cells. And so here we have 3 different stains. So this bacteria was labeled with 3 different fluorescent chemicals all at once. But then 3 separate fluorescence images are taken, separately, each with a different set of filters. And then those 3 images are superimposed on top of each other to make this composite image showing like all 3 dyes together. This is a different 2 stain microscope image that has a red stain and a green stain. The red stain is used to, will attach to cell membranes that are intact, whereas the green stain will permeate through cell membranes for cells that are dead. And so this is an assay that's done to measure, like out of a population of bacteria, how many are dead. And so this is used to test, for example, if we have some antibacterial treatment, we want to know how effective it is. We would use fluorescence in a live dead stain like this to tell like how effective our antibiotic was or our decontamination procedure. Here's another kind of cool 3 wavelength fluorescence image. This is a fluorescence image of a slice of a rat's brain where different fluorescent dyes are used to attach to different parts of different cells within the brain and also different parts of cells within the brain. So here the blue stain will concentrate itself into the nuclei of neurons. Red will stain only neurons, and then green will stain the capillary lining cells and the microglia. And so in this way, it's possible to see the structure of this tissue and to kind of see how the nuclei, neurons, and microglia are arranged amongst themselves. So those 3 images that I showed you all come from a fluorescence microscope, but some of those microarray things that I talked about earlier are imaged in a different way with something called a microarray fluorescence scanner. And so in a fluorescence scanner, rather than taking a microscope image, we take a single laser and we focus it down to a very small point, and then we take that laser and we can raster it across the surface and point by point illuminate the whole surface of a glass microscope slide. So if I have a glass microscope slide here that has a bunch of different fluorescent spots on it, I can take my laser and scan it up and down, gradually working our way across. But then for each point, we measure the fluorescence output with a photomultiplier tube that just measures simply intensity. So in order to build an image, we actually take our photomultiplier output point by point and put that together to make something that looks like a microscope image, but really it is not. But just like before, we have an emission filter that removes a laser wavelength from our photomultiplier tube. And then we, instead of a xenon arc lamp, we just use a laser, single wavelength going in, so no need for any other filters there. So, let's see, I think I have 10 minutes.
Yeah, you are just close to 5 minutes.
5 minutes, okay.
Yeah, I mean, if you want to give a few minutes for them to ask you questions.
Okay, maybe I'll talk about the DNA microarray, then I'll stop after that. So a DNA microarray, as I mentioned at the beginning, is a single glass microscope slide that has millions of individual spots on it that are printed with a special printer that can put picoliter size of droplets of DNA into an XY or grid of spots. Each spot has DNA with a different sequence representing a different gene that may be turned on or turned off in a specific tissue. So in a DNA microarray experiment, you have your device surface, your spotting machine, your printing machine will make a million individual spots, each one with a different DNA sequence. And then that whole array will be exposed to DNA that comes from a sample that can be derived from cancer cells or healthy cells from a particular tissue. That DNA that comes from the tissue will be labeled with a fluorescent dye. And using a laser and a fluorescent scanner like that, you would excite the fluorescent dye and then measure with a photomultiplier tube the intensity, the fluorescence intensity that comes from a particular spot. And so if our sample happens to have like no matching DNA, no complementary DNA, then we'd see a dark spot here. If our test sample has matching DNA that matches a sequence of DNA that's on our microarray, then we'd see a bright spot come out here. So a DNA microarray experiment is typically done like this, where we take 2 samples. One might come from the same person, one might come from a tumor, one might come from healthy cells from the same kind of tissue. So in this kind of experiment, the scientist is asking what genes are turned on or turned off that are different between a cancerous tissue and a healthy tissue. So the DNA is extracted from the tissue. I won't talk about how we do that. But then the DNA from the healthy tissue might be labeled with a red fluorescent dye. The DNA from the cancerous tissue might be labeled with a green fluorescent dye. These will be mixed together and exposed to our DNA microarray that has those million capture spots of DNA sequences on them, and then we would see kind of which spots end up with green DNA on them, which ones end up with red DNA on them, and which ones have both. And so ones that have both, that would represent a gene that's expressed by both tissues the same way. Those aren't very interesting. However, biologists are really interested in genes that are expressed only in healthy cells or only in cancerous cells to be able to tell the difference. So what's interesting is that all the cells have the same DNA sequence. Some of the genes are turned on, some of them are turned off. We want to know which ones are important or what genes are turned on or turned off due to a disease such as cancer. And this kind of experiment is used not just for cancer, but for many, many other, asking a lot of questions, or biologists might want to know what genes are turned on or turned off in the leaf of a plant versus the stalk of a plant, for example. Okay, so I think because of time, I'll stop there. So I'd be happy to answer questions about fluorescence. I didn't get to the part about my research, but perhaps for another time. Thank you.
We have some time for discussion, maybe 15 minutes or so.
Yes.
If we attach fluorescent molecule to our target or bacteria, does it affect the behavior or the property of the protein molecule?
Yes, so in fact that's a really great question. So as we attach, as we attach fluorescent dye molecules to a bigger molecule like a protein, like I mentioned, there's no way to control exactly where those fluorescent dye molecules are going to end up. Now the way the protein molecules work is by like a mechanical lock and key kind of fit, where proteins interact with another protein by the way that they physically, mechanically come together. So if a fluorescent molecule happens to stick to or interact with the active or important parts of a protein molecule, then it can affect the way they interact. So people do worry about that. For DNA, it's actually pretty easy to attach to engineer fluorescent dye that will only attach to parts of the DNA molecules that are not active in their complementary binding. For antibodies, it's also possible to do that, but it's not possible in general. So that's one. So it's called an epitope blocking. If a fluorophore happens to mess up with the way a protein interacts with another molecule, and that's actually a big advantage of label-free biosensing methods, is that they're not subject to that kind of problem. Yes?
So you mentioned there are several things the excited electron could do to come back to its ground state, right?
Yes.
And we would prefer it to be for it to give out a photon. So are there ways to ensure that it goes in that direction as opposed to, say, phosphorescence or internal conversion, or is it just up to chance?
So I don't completely know the answer to your question, other than to say that different fluorescent dye molecules can have very different fluorescent yield or quantum efficiency. And so when chemists engineer these molecules, and that's how all these molecules actually come into being, is that chemists play around with different chemical functional groups. They kind of add something here, subtract something there, and then physically test them out. They try to engineer something that will end up having high quantum efficiency because that's more desirable. So why does one molecule have better quantum efficiency than another? That I'm not completely sure about. But I know that like the likelihood that molecules have, or the electrons have a more or less clear path of oscillation without having collisions with the nuclei is important. And so a lot of times we see fluorescent molecules that are efficient be like long, skinny molecules rather than more like balled up molecules. And so and then also their ability to withstand chemical reaction. So like those oxygen functional groups that I showed on there, those are not desirable. So a lot of times people will engineer molecules that have less photobleaching by removing as much oxygen as possible. Yes?
You have mentioned that for the organic dye, there's a problem of
Yes. So, so one example is kind of like what I've shown here with the DNA microarray. By having a control, it's possible to, like an experimental control, it's possible to control for the fact that photobleaching is taking place. So in this kind of experiment, for example, the absolute fluorescence intensity is not what matters, but rather it's the ratio of fluorescence intensity between healthy tissue and the cancer tissue, between the experiment and control. So, so if they're both undergoing the same amount of photobleaching, then the quantification is easier. Other thing is that with this kind of experiment, the measurement is taken one time. So whereas with like imaging cells, you might want to actually look at the cells or see them swimming around or interacting or doing something. You have to hope that you can see that happen before everything goes dark. But with these kinds of measurements, it's only, you know, the microarray slide is only scanned one time. So whatever photobleaching happens after that one time, you don't care about.
So I'm asking about for the cell imaging rather than experiment, how does people solve this problem? That they are using a fluorescence dye to maybe take a photo or something.
Right. So so sometimes you might have to take like a photo quickly, or you can choose fluorescent dyes that have longer, are able to withstand photo bleaching for longer. There aren't any that have no photo bleaching, but there's some that last longer than others. One thing that's becoming more commonly used in cell imaging are things called quantum dots. I'm not sure if anyone's heard of quantum dots in this group, but rather than a quantum dot is a tiny semiconductor particle that gives off light, much like a fluorescent dye does. But since it's not a chemical, it's a tiny semiconductor, they don't photo bleach. And that's their major advantage, is that you can put them in cells and look at them for as long as you want, and they won't get dimmer. There's also people to try to increase the amount of time that they can look at things before photo bleaching. Might use a cell media that's specifically made to reduce rate of photo bleaching. I'm not sure what's in there, but there's actually some cell media that are like marketed to be to slow down rate of bleaching. Yes.
So why not use quantum dots all the time? What's the disadvantage? Why use?
Well, for quantum dots. So that's a great question too. So why not always use quantum dots? Well, fluorescent molecules are really small. So they're comprised of like a small group of carbon atoms and sulfur and oxygen, but it's a molecule. They're tiny. A quantum dot is actually substantially larger. Their diameter is between 5 and 10 nanometers. That doesn't sound too big, but it's huge compared to a molecule. So it's harder to get quantum dots to go into through the cell membrane or through into the nucleus. You kind of have to develop tricks and ways of chemically modifying them so that they can get into different parts of the cell. So it's still a research area, but they are becoming more and more popular compared to fluorescent dye. But I would say fluorescent dye still rule the world of cell imaging by a lot. Yes?
So do they have different absorptions and emissions like different dyes, or all quantum dots are pretty much the same absorption and emission?
So quantum dots, there's quantum dots for all ranges of color. So you can get them for any wavelength you want, from ultraviolet all the way through infrared. They even sell them in cool sounding colors, like it's not like the colors that you buy cars in. So they're like emerald blue or lapis something, or they have like hues of color, so you can get whatever you want. Yeah. But the color is engineered by the material the quantum dots are made of and the size of the dot. Yes?
I'm curious about the protein microarray. And you label the fluorescent on the samples protein, or you will use another antibody labels fluorescent to detect the sample?
Okay, so maybe when I go ahead, I'll go to my protein microarray thing here, but I'll skip all this. I have a thing here. Okay, so for a protein microarray, similar to a DNA microarray, except that instead of having a capture DNA molecule, you'd have a capture antibody. They'd be applied in spots with the same kind of spotting method. Then we would expose that to the blood sample, and the blood sample would have the biomarker protein in it, the thing we're trying to detect. So that would specifically interact or be captured by the capture antibody. Then in order to see it, we have a second detection antibody. So this is called a sandwich assay, where the detection antibody is like the upper layer of bread, and our biomolecule we're trying to detect is like the bologna, or peanut butter jelly, or whatever. But then the detection antibody is what would have the fluorescent dye on it. And so we actually detect the fluorescent dye that's attached to the secondary antibody. Does that answer your question for a protein microarray?
So how about the sensitivity of the protein microarray compared with the maybe ELISA kit or
Oh, okay.
others?
Oh, so you're getting me to go to all my good questions. So this is some of our work with protein microarrays, where actually we're using a device called a photonic crystal. I did talk about it, to amplify the output by like 2 to 3 orders of magnitude. But we're able to get down to concentrations on the order of tens of femtograms per ml protein concentration in serum. And so that is much higher than ELISA, which is a couple orders of magnitude higher than that. Yes?
Can we control and precisely control how many, for instance, molecule to attach a protein when we need to determine ultra trace penetration and conductivity?
So there are, I only showed one example of how you might covalently attach a fluorescent molecule to a protein, but there are many different ways. So the way to control it is to actually know something about the molecule you're trying to label. So for example, if you have a protein, if you know the amino acid sequence for that protein, which is known for most proteins, you'll know in advance how many NH2 functional groups will be on the outside. And so you'll know approximately, you'll get like one fluorescent molecule for each NH2 functional group on the outside of a protein. And so you can, to that extent, know how many proteins will end up on your molecule. However, that's not the same as controlling. So I wouldn't say that it really is strictly controlled that way, unless you're somehow able to like label your protein at a really low, using a really low concentration of dye or something, so as not to occupy all the available binding locations. Questions?
One slide is talking about the cell life and death, and how the dyes were going to how the dye will distinguish which one is the life, which is the death. What mechanism the dye will go to different the way you want it to?
Again, I'm not an expert at that either, but cells that are dead have more permeable cell membrane, and so it's easier for a molecule to get through the membrane and get into the inside of the cell. And so in choosing like a live dead stain, the dyes might be chosen on their size or molecular weight, where the cell membrane acts like a filter that allows smaller molecule to go through. The bigger molecule stays outside and ends up binding with outside part of the molecule. The proteins can also, the fluorophores can also be prepared with functional groups that allow them to attach with specific other molecules. So there's some that are like DNA binding fluorophores that have a higher affinity for nucleic acids than other molecules. And so that selectivity may not be perfect, but it may be good enough to get the contrast in the
Okay, in the interest of time, let's thank Professor Cunningham again. To appreciate your lecture, we present you a great class.
Thank you very much.
Okay.
Thank you.
And take a picture. Sorry. Don't photo bleach us. and also our Taiwanese colleagues made a t-shirt for you.
Oh, thank you. Thanks very much.
Thank you very much. Thanks. Now, we are